ആ യൂട്യൂബിൽ ഇട്ടിട്ടുണ്ട് തോന്നുന്നു ലൈവ് സ്ട്രീം.
Am I audible now?
Yes, ma'am.
Okay. So regarding the absorption, which all are the transporters? We talked about the GLUT, sodium dependent glucose transporter, and sodium independent. And sodium independent is by which mechanism? It is facilitated diffusion, and it is by ping pong mechanism. Okay. Which is the one insulin dependent?
GLUT4.
GLUT4. It is present in mainly in
Skeletal muscles and
adipose
tissue.
adipose tissue. Okay. Which is the one which is present in liver, pancreas?
GLUT2.
GLUT2. It is a high affinity transporter.
Low affinity.
Okay. High affinity transporter is
GLUT3.
which is the one specific for fructose?
GLUT5.
GLUT5.
GLUT5 is for fructose. Okay. And so we have finished with the digestion and absorption of carbohydrate. Now we are moving on to the disorders associated with the digestion and absorption. Okay. Defects in digestion and absorption of carbohydrate. Of this mainly we are studying. Can you see the slide?
Yes, ma'am.
Okay. So we are studying about the disorders associated with the digestion and absorption. The main one is the lactose intolerance. So for the digestion of the lactose, we need the enzyme lactase. When this lactase is deficient, the lactose cannot be digested. Okay. So this lactose intolerance, it can be a congenital genetic variety, or it can be an acquired cause. The congenital one, which means it is present from the birth, it is a rare disorder and it is seen in infants. And what is the feature is that the infants or the newborns cannot tolerate milk. As soon as they drink milk, there will be they suck the milk, there will be difficulty and digestive problems, there will be vomiting, diarrhea, etc. Okay. Acquired causes are also there. It is due to physiological, there is a physiological decline in the enzyme activity with age. That is physiological thing, which means that when we in the newborn or infant stage, they depend more on milk, mother's milk. So the enzyme activity needs to be high. After that, we will change our eating pattern to carbohydrate and like cereals, pulses, etc. So with age, there is a physiological decline in the enzyme activity. So lactose intolerance, acquired causes can be simply the physiological decline, or there is a sudden change to a milk-based diet. So when we shift the milk-based diet to the other diet, there will be a physiological decline. After that, if we switch again to a milk-based diet, initially there will be an intolerance. Okay. Or there can be injuries to the mucosa. The intestinal mucosa is the site where the disaccharidases are present. So when there is an injury to the mucosa or there is an ulcer, the enzyme producing parts in the intestinal mucosa gets eroded, so that there will be a decrease in the lactase enzyme, resulting in the lactose intolerance. Is it clear?
Yes, ma'am.
Yes, sir.
Okay. So there will be inability to hydrolyze the lactose. The absorption will be impaired. But these lactose will be present in the digestive tract, and that will be fermented by the bacteria. The bacterial fermentation of the lactose within the intestine can produce gas, and osmotically active solutes can draw water into the intestinal lumen. So this gas production and osmotically active solutes which are produced as a result of the fermentation, they will draw water into the intestinal lumen from our tissues. That can result in the diarrhea and abdominal cramps. So gas production by the fermentation is the cause for flatulence. Okay. How will you diagnose is that in the stools, normally this carbohydrates, lactose, all will be digested and absorbed, nothing will be excreted through the stools. But here the lactose will be excreted. So when stool sample is taken and Benedict's test is done, it will be positive. What can be the treatment? Milk cannot be digested and absorbed. So the withdrawal of the milk is the treatment, and instead of milk we can take curd or yogurt. Okay, that will be already fermented thing, so it can be absorbed. And there will be pre-treated milks available which already contain the lactase. That will itself digest the lactose into its component so that it can be absorbed. Is it clear?
Yes, ma'am.
Yes, ma'am.
By this, we finishes the digestion and absorption of carbohydrates, and we'll move on to the metabolism proper. Okay, moving on to the carbohydrate metabolism. Of this, some of the topics you know very well, like glycolysis, right?
Yes, ma'am.
Yes, ma'am.
Okay. So today we are discussing about the glycolysis. So it will be a discussion only. You have to answer. Okay. Regarding the metabolism of carbohydrate mainly means it is the central molecule in the carbohydrate is the glucose. So we are concerned more about the glucose, the pathways in which glucose involved are more discussed. So the first pathway is the glycolysis. It is otherwise called as EMP, Enten-Meyerhof-Parnas pathway, based on those who discovered it. And we can call it is the only metabolic pathway occurring in all cells. Why? Because the site is, where is the site of glycolysis?
Cytoplasm.
It's in the cytoplasm. So all the cells will have definitely cytoplasm. So this glycolysis is the only metabolic pathway occurring in all cells. And it can function in both aerobic and anaerobic condition. You know the products of anaerobic and aerobic glycolysis are different. You'll see it again. And the erythrocytes or RBC, they does not have most of the organelles. Okay. For the ETC and energy production, which organelle is important?
Mitochondria.
Erythrocyte does not, RBC does not have mitochondria. So they depend only on the glycolysis for their energy. So it is a major pathway that is occurring in the RBC. And the muscles after contracting for a long time, a few minutes, they lose all the stored energy. They will lose all the sudden energy. The aerobic, the oxygen part will be lost. So it will depend upon the anaerobic glycolysis for the muscles which are contracting. Okay. So this EMP pathway is called as the preliminary step for the glucose metabolism. First, the glucose is converted through the glycolytic pathway. It is converted to pyruvate. This pyruvate by a link reaction is converted to acetyl-CoA. So initially, glucose will be converted to pyruvate. This is what happens in glycolysis. If it is anaerobic one, it is converted to lactate. What happens to the pyruvate? Pyruvate by a link reaction, you will study, I will tell you. It is converted to acetyl-CoA. It is this pyruvate is 3-carbon compound. It is converted to 2-carbon acetyl-CoA. This acetyl-CoA is the one which is entering the TCA cycle and finally entering the electron transport chain to produce the ATP. So it is this glycolysis which is the preliminary step for the glucose metabolism. The final oxidation occurs in the ETC. For that, this glycolysis is the initial preliminary step. Is that clear?
Yes, ma'am.
Okay. This carbohydrates, monosaccharides like fructose, galactose, all other carbohydrates are metabolized through this glycolysis only. And all the enzymes, as I told, it is seen in cytoplasm.
മാം ഇത് സ്ലൈഡ് കാണിക്കുന്നുണ്ടോ മാം? നമുക്ക് കാണാൻ പറ്റുന്നില്ല.
കാണുന്നില്ലേ? സ്ലൈഡ് കാണിക്കുന്നില്ല.
ഇല്ല മാം.
സ്ലൈഡ് ഉണ്ടായിരുന്നോ? മൈ ഗോഡ്.
എഴുതുന്നതും കാണുന്നില്ല, സ്ലൈഡും കാണുന്നില്ല.
ഇല്ല
മാം, ഒരു വൈറ്റ് ഇത് മാത്രം കാണാം.
Okay. Let me see. Can you see now?
Yes, ma'am.
Yes, ma'am.
Just once more, I will rush through the slides. Okay. Carbohydrate metabolism. So glucose is a central molecule. It is always also called as EMP pathway. Only metabolic pathway. It is only metabolic pathway occurring in all cells. Function in both condition, aerobic and anaerobic condition. Provide energy for the RBCs because it does not have the mitochondria. And anaerobic glycolysis is the one which provide energy for the muscles during exercise. It is a preliminary step for the glucose metabolism. That is the thing I discussed just now. The glucose is the one which is first converted to pyruvate. If it is an anaerobic thing, this pyruvate is converted to lactate. What happens to the lactate pyruvate is that pyruvate is oxidatively decarboxylated by pyruvate dehydrogenase reaction. We will study in the coming classes. It is converted to acetyl-CoA. It is this acetyl-CoA that enter into the TCA cycle or the citric acid cycle. From the TCA cycle, for the energy production, there is an entry into the electron transport chain. The energy currencies like NADH, FADH, all these things enter into the electron transport chain to finally produce the ATP. So for the glucose metabolism, finally we'll get ATP. For that, this glucose to pyruvate or the glycolysis is the preliminary step. Is it clear?
Yes,
Yes,
ma'am.
ma'am.
Okay. The other monosaccharides like fructose, galactose, all are metabolized to the glycolysis. And all the enzymes are located in the cytoplasm. So we should see the glucose entry into the cell. Glucose entry into into the cell is mediated by certain transporters. We studied about the GLUT4. GLUT4 is the one which is present in skeletal muscles,
Heart.
And adipose tissue.
and adipose tissue.
And cardiac tissue.
Cardiac muscles also. Okay. And so this, with the help of these transporters, the GLUT4 is also under the influence of insulin. So it helps the transport into the cells or the tissues. So availability of the glucose in the cells is increased by the entry into the cell, and it is by the GLUT4 these glucose enter into these tissues. It is under the influence of insulin, and GLUT2 is the one which facilitates the entry of glucose into the liver. But you have to remember it is independent of insulin.
Ma'am, audio കേൾക്കുന്നുണ്ട്.
Okay, okay. ഞാനൊന്നും പറഞ്ഞില്ല.
Okay, ma'am.
Okay, so we will move on to the steps proper for the glycolysis. So this is the step 1 of glycolysis. It's a ring form of glucose. The first enzyme to act is the hexokinase or glucokinase. Both can act on the glucose. So kinase means it is the addition or removal. Kinase means addition or removal of the phosphate. So here it is the addition of phosphate. It is this phosphate added to the 6th carbon of the glucose to form the glucose 6 phosphate. Okay. This is the first step. For that, it needs magnesium ions. For the enzyme hexokinase to act, it needs an enzyme. It needs a metal ion, which is magnesium. You'll study this under the enzymology. It will be, I think, tomorrow you'll start the enzymology class by Dr. Meera. So she'll explain what is the role of metals in the enzyme. For the enzyme action, the enzyme, complex enzyme will have a protein part and a non-protein part. This part is called coenzyme. It can be a vitamin derived or it can be a metal ion. So some metal ions enzymes. So this is a complex enzyme. You will study it in under enzymology. It needs one metal ion, which is the magnesium. Okay, magnesium is the one which is needed for the hexokinase, and it needs energy also. ATP, one molecule of ATP will be utilized, which means one ATP will be converted to ADP for the conversion of glucose to glucose 6 phosphate. Clear?
Yes, ma'am.
Okay. From the glycogen also, you will study under glycogen metabolism. I will teach you. The glycogen will finally get converted to glucose 1 phosphate. That can also enter into the glycolytic pathway to reach the to form the glucose 6 phosphate. You will study under the glycogen metabolism. Okay. I told you 2 enzymes are there, hexokinase, glucokinase. Both can act on the glucose to produce the glucose 6 phosphate. Let's see the difference between hexokinase and glucokinase. This thing I think you're not familiar, right?
No, ma'am.
This is important for you. You have to study the difference between hexokinase and glucokinase. Hexokinase is the one which is present in all cells, whereas the glucokinase is present only in liver. And you have to know what is Km value. Do you know?
Yes, ma'am.
Yes, ma'am.
What is that?
Michaelis-Menten's constant.
Maximum
Okay, so
Concentration.
Maximum velocity at concentration.
velocity, half of maximum.
half of maximum.
concentration is half
Concentration of
maximum velocity. How much is the substrate concentration when the velocity is half of the maximum? The reaction is occurring at half of the maximum rate. What is the substrate concentration? That is called the Km value. You will study under the enzymology. Dr. Meera will teach you. And you can note that the Km value is very low in the case of hexokinase, whereas it is high in the case of glucokinase. The Km and affinity are inversely proportional. So the Km value is high for glucokinase, which means that the affinity will be very low. So when high concentration of substrate, which is a substrate, it is a glucose. Only when high concentration of substrate is present, this glucokinase will act in the liver. Okay. The action of glucokinase in the liver is that the excess of glucose has to be converted to glycogen. For that, the glucokinase has got a role. So this thing, Km value is low for hexokinase and affinity will be high. In the case of glucokinase, it is low affinity and high Km value. Specificity, hexo, as the name indicates, it is common for all hexoses. All hexoses will be acted upon by the hexokinase, whereas only glucose is acted upon by the glucokinase. And induction also you will study under the enzymology. Dr. Meera will teach you. There is no induction in the case of hexokinase, but the glucose or insulin, when present, it will induce the enzyme. Okay, the enzyme will be induced or more produced in the case of when glucose or insulin is present. Under the influence of glucose or insulin, the glucokinase will be induced. The function is in the case of hexokinase, it is involved in the regular metabolism, glucose glycolysis. The glucokinase role is, I told you, when there is high glucose level, as a result of that, if the body metabolism is normal, the body will produce insulin. Insulin will be secreted by the pancreas. Under the influence of insulin, why? Because insulin is an inducing agent, induction. Insulin, under the influence of insulin, the glucokinase will be more produced. As a result, what happens? It is present only in liver. Okay, the glucose, this glucose 6-phosphate entry will be there in the liver, and from the liver, from the glucose 6-phosphate, this glycogen will be produced. Excess glucose in the blood will cause the disease called diabetes. So the blood glucose has to be regulated. It is with the help of conversion of glucose. One method is conversion of glucose to glycogen, which occur in the liver. That is mediated by the enzyme glucokinase. That is induced by two things: glucose, the substrate, and the hormone insulin. Is it clear for you?
I mean, hexokinase muscle cells.
Or all cells, all.
Okay, ma'am.
Okay, it is involved in regular metabolism. All cells in glycolysis help in the hexokinase, whereas this glucokinase is present only in the liver. It is for the conversion of glucose to glycogen when there is high glucose level, which is induced by the insulin. Clear?
Ma'am, one doubt undo. Appo ee hexokinase liver lundo?
Hexokinase liver. Avadeyum glycolysis. Okay.
Ma'am, audible alla.
Voice break out.
Hexokinase liver. Because for the glycolysis to happen, it needs hexokinase. Mostly this glucokinase is concerned with the conversion to this glycogen. Both will be present in the liver. Am I audible now?
Yes, ma'am.
Okay, shall I move on?
Yes, ma'am.
After the hexokinase or glucokinase reaction, the product formed is glucose 6-phosphate. There are different fates for glucose 6-phosphate. You'll see. First is the glycolysis. For the glycolysis, it is an intermediate. Then this glucose 6-phosphate is also needed for the gluconeogenesis. What do you mean by gluconeogenesis?
Production of nucleus.
Production of
Formation of glucose.
Production of
Glucose from other substrates.
from like
other substrate.
Yes,
We'll
yes.
see it in the coming classes. So from neo means new, neogenesis. So formation of glucose from new other sources is the gluconeogenesis. For that also the glucose 6-phosphate is an intermediate. Then for the pentose phosphate pathway we'll study. For that also glucose 6-phosphate is an intermediate. Glycogenesis that we see, we'll see. Glycogen synthesis. Genesis means synthesis. Glycogen synthesis from glucose. For that also glucose 6-phosphate is an intermediate. That is about the role of the glucose 6-phosphate. The second reaction, it is the this is the first reaction itself. The second reaction is the phosphohexose isomerase. Okay, this glucose 6-phosphate is converted to fructose 6-phosphate. Okay, the difference between the ring furanose and pyranose ring. Okay, this is the furanose ring, 5-membered ring. This is a 6-membered ring. So conversion by an isomerase reaction, the glucose 6-phosphate is converted to fructose 6-phosphate. You can see the reaction is really reversible. Okay. Coming to the third step, glucose 6-phosphate is converted to fructose 6-phosphate in the second reaction. Third reaction is the fructose 6-phosphate, another kinase, which means there is an addition or removal of the phosphate group. So you can see here one more phosphate group is added. Okay. So this is the first carbon. This is the 6th carbon. Okay. Already at the 6th carbon, there was a phosphate 2. One more phosphate is added to the first carbon to form the fructose 1,6-bisphosphate. So it is a kinase reaction. So from where this phosphate, extra phosphate come? It is from ATP. Another ATP is that ATP is utilized, ATP converted to ADP. As it is a kinase, for the reaction it needs coenzyme. It is a metal ion, that is magnesium. Okay, so phosphofructokinase reaction. As you saw, it is an irreversible reaction. The second reaction you saw it was reversible. There was a double-headed arrow. Here, only one arrow, means it is an irreversible reaction, and it is called as a rate-limiting reaction. One word question can be asked like, which is the rate-limiting enzyme in the glycolysis? That is the phosphofructokinase. There will be 3 irreversible steps for glycolysis. That 3 enzymes are the key enzymes of glycolysis. But among the key enzyme, which is called the rate-limiting enzyme, it is the phosphofructokinase. Okay. It is a key enzyme. There are 3 key enzymes. Among the key 3 enzymes, it is the phosphofructokinase, that is the rate-limiting. This word is important. Key enzyme is important. Okay. How is this regulated? Is the allosteric regulation. Don't worry, you will study allosteric regulation under the enzymology. Dr. Meera will teach. Okay.
Key enzyme select the basis step reversible
reversibility basis. Key enzyme. Okay. Glycolysis reversibility basis. Okay. All reactions are reversible. Okay, gluconeogenesis നടക്കുമ്പോൾ ഈ 3 reactions മാത്രം 3 key enzymes മാത്രമേ reverse ചെയ്യാൻ പറ്റാവുള്ളൂ. For that there is another enzyme to reverse all the pathway.
Okay, ma'am.
Okay. And it is also inducible by certain molecule. That will also you study under the enzymology. Okay. So this is the phosphofructokinase reaction. Fructose 6-phosphate to fructose 1,6-bisphosphate. Magnesium required. ATP is required. Okay. Then fourth reaction is aldolase reaction. Why it is called aldolase reaction is that the reversal needs, sorry. So this is an aldose. Okay. There is a condensation of aldose. Reversible reaction. The reversal. This way it is an aldol condensation. That is why it is called as aldolase enzyme. Okay. Fructose 1,6-bisphosphate is cleaved into two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So 6-carbon compound is broken down into 2 3-carbon compounds. They are the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay. What is the peculiarity is that this two forms, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, are freely interconvertible with the help of a trimose isomerase enzyme. Okay. So the rest of the things are concerned with the glyceraldehyde 3-phosphate. Even though dihydroxyacetone phosphate is formed, that can also be converted to glyceraldehyde 3-phosphate. So what happens to glyceraldehyde 3-phosphate is 1,3-bisphosphate. So here you can see there is only one phosphate group. Here you can see one more phosphate is there. This is the third carbon only had phosphate. To that, one more phosphate is added to the first carbon. So for that, the phosphate is provided by what?
ATP.
It is not by ATP.
NADP. NADP.
Phosphorus acid.
Phosphoric acid.
Phosphoric acid.
Okay, phosphor. It is by the phosphoric acid. Here, no ATP conversion is not involved. And for that, it needs one more thing. It is NAD. Actually, in this reaction, one NAD is converted to NADH. Okay. This NADH will enter into the oxidative phosphorylation pathway to produce
ATP.
3 ATP. 3 ATP.
ATP. Okay. How much ATP?
3
ATP.
Actually, it is 2.5 ATP, not 3. Okay. In earlier textbooks, it was written like 3 ATP, but actually 2.5 ATP is already produced. Okay. So glyceraldehyde 3-phosphate conversion to 1,3-bisphosphoglycerate. Here no ATP is involved. Phosphate group is added from the phosphoric acid, and the conversion is there is a conversion of NAD to NADH, and the NADH enter the oxidative phosphorylation pathway. Finally, enter the ETC, electron transport chain, to produce ATP. The yield of ATP is 2.5 ATP. So we have studied in the first reaction, one ATP was utilized, not produced. The third step also, one ATP was utilized, not produced. The fifth reaction we saw, actually 2.5 ATPs are produced. It is only after entering the ETC, or we can say one NADH. Actually, it is not one NADH. There will be 3 glyceraldehyde 3-phosphate, right? 2 glyceraldehyde 3-phosphate. Align.
Yes, ma'am.
So there will be 2 NADH.
NAD.
Okay. Or 2 into 2.5 ATP. We can say 5 ATP. This much clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, and we should know this thing, iodoacetate, which is an inhibitor of this reaction. Okay, that reaction will be inhibited by iodoacetate. So when iodoacetate is present, this reaction will not happen, and there will be no completion of glycolysis. Coming to the 6th reaction, so the product of 5th reaction is 1,3-bisphosphoglycerate. Another kinase will act. Kinase means it can be addition or removal of phosphate. Okay, here already 2 phosphate is there, 1 phosphate is getting removed. So 3 phosphoglycerate. The first phosphate is removed. Okay, either removal or addition, the enzyme is kinase. So actually, what is the conversion? ATP to ADP or ADP to ATP? This phosphate,
ADP,
extra phosphate is
ADP
taken
to
by the
ATP. ADP to ATP.
to form the ATP.
ATP.
So actually, how many ATPs are formed in this reaction?
2.
2 ATP will
Two
be formed.
ATP.
Why? Because after that aldolase reaction, 2 glyceraldehyde 3 phosphate is formed. From 2 glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate, 3 phosphoglycerate. So 2 ATP will be produced. From the NADH step itself, the products formed will be into 2. Okay. So 2 NADH at the 5th step, 2 ATP at the 6th step. Okay. From the 3 phosphoglycerate, next step is phosphoglyceride mutase. What is the function of mutase is that the change, it will shift the phosphate group from the one position to another. Here it was the first carbon, third carbon, 3 phosphoglycerate. From the third carbon, the phosphate group was shifted to the second carbon. So it is 2 phosphoglycerate. It does not need energy. 8th step is by enolase. The enolase reaction is important for you. There is an inhibitor which has got clinical importance. So 2-phosphoglycerate is converted to phosphoenol pyruvate. Enol means there is a double bond is formed. Okay, double bond is formed. This is the phosphoenol pyruvate, and this is by the removal of water. And enolase also enzyme requires magnesium. Okay, and fluoride is a non-inhibitor of this enzyme enolase. Why? How this fluoride inhibit this enolase is that fluoride will remove the magnesium ions from the medium. Magnesium ion is essential for the activity of the enolase, so that the enolase will not work. There is a clinical importance for this fluoride. For the glucose determination, blood glucose measure, the sample will be taken in a tube which has got gray cap. Okay, blood collection bottle will have got a gray cap. Gray cap bottle means it contains fluoride. We are adding fluoride tubes. We are taking fluoride tubes for the measurement of the glucose in the blood. What is the use is that? Suppose you are taking a blood sample and measuring the blood glucose level. Okay, are you listening?
Yes, ma'am.
Yes, ma'am.
Okay, blood glucose. For the measurement of this blood glucose level, you are taking sample at 9 a.m. Actually, at that time, the blood glucose level, say it will be 110 milligram per deciliter. But we took the sample after few hours or few as time passes, what happens is that the blood will contain RBC. RBC needs energy. So RBC will depend upon the glycolysis for its energy. So within the test tube, the glycolysis can happen. And after glycolysis, what is happening? The glucose is getting converted to pyruvate, right? So glucose level falls. So at the time we determine we'll get falsely a low value like 80 or something. Okay, you understood the point. So we'll get
Yes,
a
ma'am.
wrong value.
Ma'am.
If we are
Yes,
adding the fluoride, this reaction, the glycolytic pathway will be inhibited, so that the glucose will get the actual glucose value. So this is the important clinical importance of the glycolysis. We are using fluoride containing tubes to take the blood sample for glucose determination. Why? Because fluoride is an inhibitor of enolase. Actually, it will deplete the magnesium ions from the medium, so that the enolase cannot function. So 2-phosphoglycerate cannot be converted to phosphoenol pyruvate and glycolytic pathway will be inhibited.
Ma'am,
It is an irreversible inhibition by fluoride. It cannot be reversed. So it is used as a preservative for the blood glucose estimation. The next step is the phosphoenol pyruvate. Phosphoenol pyruvate is converted to pyruvate. So here it is by an enzyme kinase. Kinase means addition or removal of the phosphate group. You can see it is the removal of the phosphate group. That phosphate group, removed phosphate is taken up by ADP to form the ATP. So here also actually 2 ATP will be produced. Okay. This
Ma'am,
pyruvate
ma'am,
in
ഒരു ഡൗട്ട്. അപ്പോ നമ്മൾ ഒരാൾക്ക് ഫ്ലൂറൈഡ് കൊടുത്തു കഴിഞ്ഞാല് അയാളുടെ ബോഡിയിൽ ടോട്ടൽ ഗ്ലൈക്കോളിസിസ് നിൽക്കുമോ?
അത് ഫ്ലൂറൈഡ് പോയിസണിങ് ആവുമല്ലോ.
ഓക്കെ ഓക്കെ.
അത്ര എമൗണ്ട് ഓഫ് ഫ്ലൂറൈഡ് എത്തണം, മൊത്തം ഇൻഹിബിറ്റഡ് ആണ്. Then, so 2 ATP will be produced. This pyruvate can occur in 2 forms, enol form and keto form. That is not important for you. So 2 forms will be there. Just know that. Okay. So pyruvate.
And previous slide on the
And the
previous slide on the
okay.
ma'am,
Ma'am,
then the addition of fluoride will also inhibit the first reaction, right?
First reaction, it is not written in textbook, I think.
Ma'am, because magnesium is involved, right?
Yeah, but only the pyruvate, that reaction is only mentioned. Enolase reaction is only mentioned.
Okay, ma'am. Thank you.
I'll check one. Enolase is only inhibited. The mechanism is actually it is depleting the magnesium. I think rather than magnesium, the other ion, some other manganese, I think that can also catalyze that reaction, first and third reaction. So that won't
Okay.
be a matter. Even though magnesium is depleted, that is not a matter.
Okay, ma'am.
I'll once again check and tell. Okay. So then, what happens to the pyruvate if it is an anaerobic cycle? The pyruvate will be converted to lactate. For that, we need one NADH is used. So from where do we get this NADH?
ബോഡിയിൽ എടുക്കും.
Glycerol.
Already produced.
Glycerol.
Aldehyde 3-phosphate 1,3-bisphosphoglycerate.
3-phosphate dehydrogenase reaction produces NADH. That NADH will be used by this pyruvate to form the lactate. So here actually 2 into 2.5 ATP will be produced. Means 5 ATP will be generated. But if it goes on to anaerobic cycle, this 5 ATP will again be lost. Clear?
Yes, ma'am.
Yes, ma'am.
Okay. That is the same thing I told you. Fifth reaction, which is a glyceraldehyde 3-phosphate dehydrogenase. For that, there is an NADH production. That NADH will be utilized to form the lactate in the case of anaerobic glycolysis, which means that it is a less energy yielding process compared to the aerobic glycolysis. Okay, coming to the energetics. Shall we stop here? It's time 9.
Yes, ma'am.
We have another class, ma'am.
Next line.
Okay. So I'll continue on Thursday. Read and come. I'll ask.
Okay, ma'am.
And latecomers will
Okay,
not
ma'am.
be entertained. I'll mark absent. Thank you.
Thank you.
Thank you,
Thank you,
ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
ഇപ്പൊ ഇതിന്ന് ഇറങ്ങിയാ പിന്നെ കയറാൻ പറ്റില്ല.
ഈ സെയിം ഗ്രൂപ്പിൽ തന്നെയല്ലേ മീറ്റിംഗ് ഉള്ളത്?
അതെ അതെ തന്നെ.
അടുത്തത് ഏതാ?
Kavita ma'am.
ആ
നമ്മൾ ഈ ക്യാമറ ഓൺ ആക്കിയിട്ട് സ്ക്രീൻഷോട്ട് നോക്കുമ്പോ തിരിയോ?
ഓ മനസ്സിലാവുമെന്നോ?
തിരിയോന്ന് വെച്ച് മനസ്സിലായി.
ഇല്ലായിരിക്കും അല്ലേ?
You can actually test it, you know.
Good morning, all of you.
Morning, ma'am.
Morning,
Good
ma'am.
morning, ma'am.
Can you hear me?
Yes, ma'am.
Yes, ma'am.
Only 88 participants. Why?
I think
Because only the 100
Total 100
participants can
പറ്റത്തുള്ളൂ.
enter.
Ma'am, the maximum capacity is 100.
Again 100. Why?
others
so
are watching
We
through
all
YouTube.
guys are watching through YouTube.
Okay. Jisha or Liji in this group, Zoom. Jisha. Okay, 100 വന്നില്ലല്ലോ അപ്പൊ. 100 എന്താ വരാത്തെ? If 100 students can enter, only 90.
And I think people accidentally left the meeting after the first class.
Accident.
Just after the first.
Okay. Okay, shall we start?
Yes, ma'am.
Yes, ma'am.
Can you see the screen?
Yes,
Yes,
ma'am.
ma'am.
Okay.
Class തുടങ്ങി.
Okay, did you read the topic in the previous
ma'am.
class?
Yes, ma'am.
Okay, did you do the assignments I have given you?
Yes, ma'am. The HLA and method.
Ah, yes, very good. Can anyone of you tell me about the HLA? Briefly, few important points about HLA. Who is going to tell? ആരെങ്കിലും വോളണ്ടറി ആയിട്ട് വരുന്നുണ്ടെങ്കിൽ can, otherwise I will call. Yes. Who's that? Harsha. Harsha.
Yes, ma'am.
Okay. Okay. You can.
Ma'am, HLA or MHC are self-antigens that help in identifying and rejecting the foreign antigens.
Yes.
They are called HLA antigens because they were first identified in the leukocyte membrane.
Okay.
They are generally seen on the surface of all body cells except RBC, and they are glycoproteinaceous in nature.
Can you please come in the video? Okay. Okay, no, lightning is not proper. Okay. Okay, you can tell.
Ma'am,
Okay. Yes. Okay, okay.
Ma'am, then functions, rejection of transplanted tissues, identification of foreign antigen,
Yes.
Presentation of the foreign antigen to the T cells for the induction of cellular immunity.
Okay.
Then classification of MHC, MHC1 and 2. MHC1 is seen on cell membrane of body cells except RBC. MHC2 on the surface of antigen presenting cells, thymus and activated T cells. Ma'am.
Yes. Okay. Okay. Yes. Okay. These much points are important for you. At least you should know these points. Okay.
Okay.
These are the most important points, and the detailed structure and other properties you will learn when you go to second year classes, when you learn pathology. Okay, pathology, microbiology, you will learn in detail about this MHC. And as far as your first year is concerned, you should know these important points. That is, I will show you few slides. Okay, we will start today's session. And first I will discuss about HLA. You can, okay, switch off your video. You can mute and, okay. Okay, so major histocompatibility complex or human leukocyte antigen. Okay, it is shortly written as MHC or HLA. They are nothing but they are proteins present on the plasma membrane of cells of each individual of species. And each individual has distinct MHC or HLA. Okay, that is the identity of the individual. Okay, and this is encoded by genes known as major histocompatibility genes, MHC genes. Where are these genes located? These genes are located on short arm of chromosome 6. Okay, that point is also important. Short arm of chromosome number 6. This gene is located on short arm of chromosome number 6. And in humans, they are also known as HLA, that is human leukocyte antigen. And you can see this is the picture of human leukocyte antigen gene and the antigen. Okay, this is the structure of the segment of the chromosome. You can see here you have the gene segments. Here, this part of the segment is called representing the MHC2 molecules, and this part is coding for MHC1 gene, 1 antigen, and this part is actually representing the complements, proteins, and others or cytokines. This part is known as MHC3. In some books, it is classified into 3: MHC1, 2, and 3. Okay, so for you, 1 and 2 are important. Okay, and this is the structure of the MHC antigen or human HLA. Can you see this? This is this is MHC2 and this is MHC1. Okay, so it is like structure and just you should know that antigen binding area. Okay, that is why the structure is important when you go to pathology. Very, very important when you go to, when you in pathology and microbiology, it is a short note for you and you should know the structure. Okay. And these are the classes, as you told. MHC class 1 is the most important thing, glycoprotein present on all nucleated cells. Okay, that is why it is absent on RBCs. Okay, almost all the nucleated cells you have MHC class 1. And the structure, you should know that there are alpha and beta chains. And here you can see in the alpha chain, there are 3 subunits, alpha 1, alpha 2, and alpha 3 subunits. And there is a groove between these 2 subunits, distal subunits, this distal part of the alpha subunit, alpha 1, between alpha 1 and alpha 2, you can see there is a groove formed. Okay, and this groove is the site for binding the antigen. Okay, that point you should know. So the distal domains are folded to form a groove, which is the antigen binding site for antigen presentation to CD8 plus T cell. That what is CD, I will tell you later. So CD8 plus T cells are also known as cytotoxic T cells. Okay, so MHC1 antigens or HLA1 will present antigens to CD8 T cells or cytotoxic T cells. Okay, So antigen presenting cells will present the antigen to cytotoxic T cells by presenting it on the MHC1 antigen. മനസ്സിലായോ? Understood that point?
മാം ഒന്നുകൂടി പറയോ?
മാം മനസ്സിലായില്ല മാം.
മനസ്സിലായില്ല. അതായത് ഈ MHC protein എന്ന് പറയുന്ന സാധനം, MHC protein is present on all nucleated cells. Okay, and which is the most important thing in immunity is the presentation of antigen to the lymphocytes. Antigen should be presented to the lymphocytes for activating the lymphocytes. Okay, so how these antigens are presented? These antigens are presented in different ways. One is by presenting with MHC class 1 proteins, and the other is by binding with the antigen presented with MHC class 2. Okay, why there is this difference is because you have two types of T lymphocytes. One is cytotoxic lymphocytes and helper T cells. Okay, cytotoxic T cells and helper T cells. We have learned in classification of lymphocytes, isn't it? T cells. You have cytotoxic T cells and helper T cells. So these are the cells which should identify the antigens. So they can identify antigens only when these antigens are presented along with MHC proteins. Understood?
Yes, ma'am.
These lymphocytes can identify foreign antigens only when they are presented along with MHC proteins. Okay. Okay. So coming to the cytotoxic lymphocytes, they are also known as CD8 T cells. They can identify the antigens only when they are presented with MHC1 proteins. മനസ്സിലായോ?
Yes, ma'am.
Okay, and coming to helper T cells. Helper T cells can identify the antigen only when they are presented along with MHC class 2 protein or MHC2 protein. Okay?
സ്ലൈഡ് മാറിയില്ല.
ഇല്ല, സ്ലൈഡ് മാറ്റാം. സ്ലൈഡ് മാറ്റിയില്ല. ഓക്കെ, ഈ പോയിന്റ് നിങ്ങൾക്ക് കിട്ടണം. അതുകൊണ്ടാണ് ദിസ് പോയിന്റ് ഇസ് വെരി വെരി ഇമ്പോർട്ടൻറ്റ്. Okay, this is very very important for understanding the role of T cells and B cells in immunity.
Could you repeat that once more?
Okay, I will show you in another slide. That is, this is the structure of MHC class 1. MHC class 1, you have this alpha chain and beta chain. The distal domains of this alpha chain is folded to form a groove where the antigen is presented. That point you got? Did you get that point?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Next, MHC class 2, the structure is like this. Here, these MHC class 2 proteins are present on all immunologically active cells. That is, all immunologically active cells, which are they? One is activated T cells, then B cells, monocyte macrophages, or other antigen presenting cells. In all these immunologically active cells, you have MHC class 2 proteins. But MHC class 1 is present on all nucleated cells. Okay, this point is also important. And MHC class 2, you can see there are alpha and beta chains. Okay, here the antigen is presented in the space between this alpha 1 and beta 1 chain. Okay, distal domains of alpha 1 and beta 1 constitute the antigen binding site, and this is for recognition by CD4 plus lymphocytes. CD4 plus lymphocytes or helper T cells. CD4 plus is helper T cells. Okay, did you get that point?
Yes, ma'am.
ഇപ്പൊ കിട്ടിയോ? Okay, I will. And MHC class 3, I told you genes code for complement components C2 and C4, and that is not much important for you. Just for completion sake, I'm telling you this MHC, there is another class called MHC class 3. Okay, MHC class 1 and class 2 you should know. And what is the significance of MHC? As she told, these are the significances. What are the HLA antigens or MHC proteins play a major role in recognition of foreign antigens and in immunity. And this has a major role in recognition of tissues as self or foreign and accepts self and reject foreign tissues. Okay, and HLA matching is important in organ transplantation. I told you this HLA is unique for each individual. Okay, so usually no two individuals have the same HLAs. So that is why the before transplantation you should do matching. Okay, only identical twins will have the same HLAs. Understood? And in other cases, you can have similar HLA molecules which can match for individuals. And in that case, you can do matching before organ transplantation. That is called HLA matching, which is very, very important. Even though the HLA matching is done and if they are matched properly, but there can be rejection. Okay. That is what I told you. That is, there can be minute variations. No two HLAs, no two individuals will have the same HLA molecules. Okay. So that is important in organ transplantation. All those things, or transplant rejection and all those things, you will learn in detail in pathology. Okay, and these HLAs are involved in many inflammatory and autoimmune diseases. So these are the significance. And there is something called MHC restriction or HLA restriction. That is the point which I told you. T cells will respond to processed antigen on the macrophages and other antigen presenting cells. APCs are antigen presenting cells only when they are presented along with the self MHC antigen. This is known as MHC restriction. Okay, self MHC, that is self individual MHC present idal matrame T cells respond cheyullu. Okay, that is called MHC restriction.
Ma'am.
Yes, you will have a lot of doubts in this. It is a bit complicated thing, I know. Yes, Jojo.
Ma'am, by the self MHC, do you mean the MHC of the individual or the
individual? That
okay,
is,
ma'am.
this is very, very important in case of transplantation. Okay. Transplant, there will be non-self MHC. That is self MHC in donor kidney MHC, that is donor organ MHC in okay, so T cells can respond to the antigens only when they are presented along with the self MHC. Okay. So, and this is called MHC restriction. And this, yes.
MHC restriction, MHC
restriction, self MHC, that is nammade MHC protein or antigen konde present ida matrame T cells respond cheyullu. Non-self MHC varuna condition nu parnal transplantation okke cheyyunna, if you do transplantation, you will have a donor kidney, alle? Donor kidney which is placed in the recipient's body, and in that kidney also you will have several MHC molecules, isn't it? All nucleated cells you have MHC molecules, MHC proteins. So the T cells in your body will respond to processed antigen only when they are presented along with the self MHC and MHC.
So processed antigen
that we will discuss later. Processed antigen has to be processed before presenting to lymphocytes. Okay, processing is nothing but the phagocytosis and digestion. Yes. Okay.
Ma'am, transplantation weak immunity.
Yes, weak immunity, and also not only that, we are also suppressing his immunity for preventing the rejection.
Okay.
Okay, transplantation, after transplantation, we are giving a lot of immunosuppressing drugs, immunosuppressants. Okay, that is why yes.
Ma'am, I'm just having a doubt. Is this MHC restriction one of the basis for like the rejection in organ transplantations from other persons?
Yes.
So this MHC like
Yes,
has got some
organ
genetic
transplantation is a vast topic. I will tell you the important points in organ transplantation in the next class. Okay. You note down the doubts in this section because you will have lot of doubts in this. I know. Because I am telling only the superficial parts of the immunity, because immunity as such is you will learn in microbiology and pathology in the second year. Okay. In between there are lot of points. I am telling only the important points because you will not understand the whole thing if I tell you the whole thing. Okay, that is why I'm എന്താ പറയാ കൺസൈസ് ആയിട്ടാണ് പറയുന്നത്. മനസ്സിലായോ? കുറച്ച് ഭയങ്കര ഡിഫിക്കൽട്ട് ഒരു ടോപ്പിക് ആണ്, ഐ നോ, ആൻഡ് യു വിൽ ഹാവ് ലോട്ട് ഓഫ് ഡൗട്ട്സ് ഇൻ ദിസ്. Please note down the doubts and ask me at the end of the session, or you can message me.
Okay, ma'am.
Okay. Okay. Otherwise, I cannot finish the topic. That okay. Sorry to tell you this because I want to finish this in 2 classes. Okay.
Okay, ma'am.
Okay, yes, ma'am.
Okay, so this is MHC restriction, and as a part of this, you can see that the cytotoxic lymphocytes or CD8 plus lymphocytes are able to kill and lyse the virus infected cells only when the antigen is presented as a complex with the MHC class 1 molecule. Here you can see. Okay. Can you see this? Here, this part is a CD4 lymphocyte, and this part is for cytotoxic lymphocyte, which is shown in the same picture. Here, you can see that CD4 lymphocytes can recognize the antigen. This part is, this blue round thing is the antigen presented with MHC class 2. This is the membrane of antigen presenting cell, which is presenting the antigen with MHC class 2. So this CD4 lymphocytes can recognize the antigen only when they are presented with MHC class 2. Okay. Similarly, CD8 lymphocytes, you can see here, this part is the antigen presenting cell and this is the lymphocyte. They can recognize the antigen only when they are presented with MHC class 1 molecule. Okay, the class 1 molecule structure we have seen. This is the groove for presenting the antigen, and this is presented to the T lymphocyte. Okay. Mansilayo? Understood?
Yes, ma'am.
Okay. Okay,
Yes,
so
ma'am.
that is the important point in MHC restriction. Okay, so that is about MHC. Okay, so this is an important component of the immune reactions, our immune system. Okay, and another important thing when we discuss about the B lymphocytes is the antibodies. What are these antibodies? Can you see this virus? which is
Immunoglobulins.
Immunoglobulins.
Yes. So antibodies are nothing but gamma globulins, proteins, plasma proteins. Yes, they are immunoglobulins produced by plasma cells. Plasma cells are
B lymphocytes.
formed from B lymphocytes on exposure to antigen. Okay, on exposure to antigen, the B lymphocytes are activated and converted to plasma cells, and they form the globulins or antibodies, immunoglobulins. They are actually glycoproteins, and they bind to, and the detailed about the immunoglobulin structure and all those things you will learn in biochemistry. Okay, very, very important. And biochemistry short note as well as physiology also, you can this can be asked as a short note. Okay, so antibodies will bind to specific sites on protein of antigen. Okay, antigens are have protein components. These protein components or the antibodies will bind to specific sites of proteins of antigen, and they can inactivate or neutralize the antigen. Okay, and each antibody is specific for a particular antigen. This specificity, acquiring this specificity, how this B lymphocytes will or T lymphocytes acquire specificity, we have discussed in the previous class, clonal proliferation and all those
Proliferation.
things. Yes,
Yes,
okay.
ma'am.
Yes. So that is, each antibody is specific for a particular antigen. Okay, and coming to the structure briefly, the detailed structure you will learn in biochemistry, and the structure you can see there are heavy chains and light chains. Okay, this is called a heavy chain. There are 2 heavy chains and you have 2 light chains. Okay, and that is 2 pairs of polypeptide chains of different sizes. Smaller one is called light chain and the larger one is called heavy chain. Okay, this is the light chain and this one is the, can you see this?
Yes, ma'am.
Okay, this is the heavy chain. Here the heavy chain is the structure which is structurally and antigenically distinct for each class. Okay, here the heavy chains are structurally and antigenically distinct for each class of immunoglobulin, whereas light chains are similar in all cases, all classes of immunoglobulins. It can be either kappa or lambda chains. Detailed lambda structure you will learn in biochemistry. I told you. You just know that there are heavy chains and light chains. The heavy chain is antigenically and structurally distinct for each class. Light chains you can have kappa or lambda for all classes. Okay, and depending upon this, here you can see the heavy chain. These chains have amino terminal end and a carboxy terminal end. This lower part is carboxy terminal. Here you have the amino terminal end. The amino terminal half of the chains are highly variable region. Variable means they are not, they will vary in each immunoglobulin. And this part is the part for antigen binding. Okay, so amino terminal end of the heavy half of the chains are highly variable, and this is called variable region. It forms the part of antigen binding site, and it is also known as FAB portion. AB for antigen binding. Okay, this variable portion is for antigen binding, and this part, the carboxy terminal, forms a constant region. Okay, this is called FC region. That is called constant region. Okay, and this constant region is the effector portion. Effector portion means it is the portion which is causing the effects. That is, it mediates the reactions initiated by antibodies. Reactions, what all the reactions are initiated by antibody? When an antigen binds to the antibody, these antibodies can mediate several reactions, including the complement activation. Okay, there are certain activation of several proteins called complement proteins. Okay, and this activation of complement proteins or complements will get activated, and they will come and bind to this immunoglobulin, and all these binding or reactions are taking place in the constant region. Okay, and that is why it is called effector portion. Okay, so the amino terminal is variable region, and that is for binding to antigen, specific antigen, and the constant region or the carboxy terminal forms the constant region. It is the effector portion which is mediating the reactions initiated by antibodies. And you can see there are some disulfide bridges. Disulfide bridges or bonds bridge the light and heavy chains, and also the heavy chains are attached to each other at the middle by disulfide bonds. Can you see the disulfide bonds? Okay, okay. Boring topic aana orangiya?
No, ma'am.
No, ma'am.
Okay, okay. So you can see disulfide bonds in the immunoglobulins. That is between heavy chain and light chain, as well as between the two heavy chains. Okay. And what are the different types of immunoglobulins that you might have learned in +2 classes?
IgG,
Yes.
IgA,
Yes.
IgM.
Classes of immunoglobulins are there. 5 classes: IgA, IgD, IgE, IgG, and IgM. Okay, and you should know the functions of each of them. If you are asked about the immunoglobulins, asked a short note about immunoglobulins, you should write down the important points of each class. Okay, that you will get from biochemistry. Okay, I'm just explaining I'm just mentioning the important points. Okay, IgA is the major antibody in secretions. So when an antigen is activating the humoral immunity, you can see functionally similar antibodies of all classes are produced. Almost all the classes of immunoglobulins are produced in different proportions depending upon the need. So IgA is the major antibody seen in secretions, that is providing the secretory immunity. And IgD is acting as the receptor for antigen on the B cell surface. Okay, on the B cell surface, you can see immunoglobulins which are developed for each antigen during the development. Okay, that is as we have seen in clonal selection and clonal proliferation, there are receptors formed during the development of B lymphocytes and T lymphocyte. In the case of T lymphocytes, these receptors are called T cell receptors, whereas in the case of B lymphocytes, these receptors are nothing but these immunoglobulins. Of this IgD class of immunoglobulins are acting as the receptor for antigen on the B cell surface. Got the point? Okay.
Ma'am.
Yes. Okay. So IgD immunoglobulins are acting as receptor for antigen on the B cells, B lymphocytes. Okay, and IgE, what is the role of IgE? IgE is the immunoglobulin, yes, which binds to mast cells and basophils. We have seen there are receptors for IgE on these mast cells and basophils. And these immunoglobulins will bind to these cells in allergies and also some parasitic infections. Especially helminthic infections, you have several allergic reactions. And these are mediated by IgE class of immunoglobulins. Okay, and coming to IgG, which is the main secondary antibody response. What is the secondary response? We will see later. So IgG is the main secondary antibody response. And it is the only antibody that can cross placenta. Very, very important point. IgG is the only antibody that can cross the placenta. Okay. And about IgM. IgM is the largest immunoglobulin. And it is the first antibody formed on exposure to antigen. That is called primary immune response or primary antibody response. Okay, so whenever an antigen is, whenever there is an infection, the primary response, you can see IgM type of antibodies to that antigen. Okay, so it is the first antibody formed on exposure to an antigen. Okay, these are some important points, and you should also know the size, molecular size, and the chains of these classes that you will learn in biochemistry. That is why I'm not, yes.
Secretory immunity.
Yes, secretory immunity I'm giving you as an assignment. Okay, that is an assignment for you. Secretory immunity, you should write down what you should refer what is secretory immunity. And secretory immunity is nothing but the immunity which is as a part of immune reaction, you are getting developing immunoglobulins, and these immunoglobulins are secreted in different types of secretions. So these are mainly seen in the mucosal surfaces as a part of immune reactions occurring on the mucosal surfaces in the gastrointestinal tract, respiratory tract, all those regions. Okay, and what is secretory immunity? You can refer textbooks and write down a note on that. Okay, that is an assignment for you. Okay,
Okay, ma'am.
നോട്ടിൽ തന്നെ എഴുതിയാ പോരെ?
Yes, നോട്ട് തന്നെ എഴുതിയാൽ മതി. Write in നോട്ട് തന്നെ എഴുതി വെച്ചാൽ it will be beneficial for you. അല്ലേ?
Could you repeat that once more?
Which one? Previous slide.
No, ma'am, the assignment which you have to.
That I will show you. Okay, so this is just a table from Ghana showing you the important functions of each class, which I have already mentioned. Okay, so IgA is here you can see IgA is the localized protection in external secretions, tears, intestinal secretion, etc. Okay, and that is forming part of the secretory immunity. And this chains also you should know that you will learn in biochemistry. That is why I'm skipping this. Okay, and the structure, almost all of them are monomers or dimers except the IgM, which is a pentamer. And there are some additional chains for IgM and IgA. Apart from this heavy chain and light chain, there are some additional chains. Okay, so that is about immunoglobulins. And what is secretory immunity? Secretory immunity, refer and write down a note on this. Okay. Next week, when you come for the practicals, you have to submit the WBC completed notes. Okay. So be ready.
Yes, ma'am.
Okay,
ma'am.
So that is about antibodies. And what are the mechanism, how these antibodies are acting? How this mechanism of action of antibodies? They can directly attack the invader. That is direct attack on the invader and can cause different types of reactions. They can cause agglutination, they can cause precipitation, neutralization, and or lysis. Okay, these are some reactions caused by antibodies by directly attacking the invader. What is this agglutination? Agglutination, you can yes, multiple large particles of antigens. Antigens, especially these antigens on the surfaces of different cells like red cells, RBCs. These cells will, these antibodies will cause binding of all these cells together. Okay, antigen bound cells will be there like RBCs. And these antibodies can, these multiple large particles with antigens on the surface of RBCs are bound together into a clump. That is called agglutination. And what is precipitation? In precipitation, you can see that there are certain antigens which will be soluble antigens. And these antibodies will combine with these antigens and they can form large insoluble particles. And thereby they will get precipitated so that they cannot do the harmful effects. They cannot harm our body, harm our cells. All these antigens will get precipitated and they will be eliminated from the body. That is called precipitation. And another thing is neutralization. What is this neutralization? In neutralization, the antibodies will bind to the toxic side portions of the antigen. Antigen le thanne toxic portions undavum which is causing the harmful effects. And that portion is bound by this antibody and it thereby neutralizing the antigen. Okay, then another thing is lysis. That is by directly attacking the membranes of cellular elements of the cells which are containing antigens. Okay, directly causing lysis of the cells and antigen, thereby lysis of the antigen. Okay, so these are the mechanisms of direct attack on the invader. And another mechanism of antibodies are by activation of complement system. I told you they can activate different complement proteins. And this in this mechanism, complement system, you can see that there are several proteins called complement proteins which are activated. And this activation can be in two ways. there are two pathways for activating complement system. One is classical pathway and the other is alternate pathway. Okay, detailed pathway you will learn in microbiology. Now you just know that there are two important pathways which are activated by antibodies. That is, they are classical pathway and alternate pathway. Okay, and activating this complement pathway, they can cause the immune reactions. Understood? So these are the mechanisms of actions of antibodies. Okay. So when you are asked about the immunoglobulins, you should write these also. What are the effects of antibodies? Okay, so that is about antibodies. So we have seen MHC, what is MHC and what are antibodies. And another thing you will come across when you learn immunity is a term called cluster differentiation. Cluster differentiation is short form is CD. We have seen CD8 and CD4, isn't it? What are these cluster differentiation? They are nothing but proteins. Protein present on the surface of lymphocytes or several cells. Okay, so in addition to the specific T cell receptors or immunoglobulins, lymphocytes also have some markers on their surface called cluster differentiation. എല്ലാം കൂടി കൺഫ്യൂഷൻ ആയോ? ഒരു ലിംഫോസൈറ്റ് എടുക്കുമ്പോൾ അതിന് മുകളിൽ കുറേ സാധനങ്ങൾ ഉണ്ടായിരിക്കും, അത്രയും മനസ്സിലാക്കിയാൽ മതി. ഓക്കെ, ഇമ്പോർട്ടൻറ്റ് തിങ്സ് മാത്രമാണ് ഞാനിപ്പോൾ പറയുന്നത്. ഓക്കെ, and there are lot of structures present on the cell membranes, lot of proteins present on the cell membrane. Okay, of these important proteins which are important especially in immune reactions are mentioned here. Okay, of these important things are MHC or HLA, then immunoglobulins, then cluster differentiation. What is this cluster differentiation? There are different markers. These they are called markers because they can be used as identifying or marking the specific cells. They can be used as markers for specific cells. Like CD4 is a marker for helper T cells. Okay, you can identify the CD4 cluster differentiation 4. When you find CD4 positive, CD4 positive, that cell is helper T cell. If the CD8 is positive, that cell will be cytotoxic T cells. And CD3 is present on all T cells. So CD3 is a marker for T lymphocyte. Like that, CD19 is a marker for B lymphocyte. Okay, these are some examples. Only some examples. Lot of cluster differentiation. Yes, proteins are there. Lot of proteins are there. Yes.
If we want to confirm whether a particular cell is helper T or cytotoxic T, prior to this, should we first confirm whether it's T cell by checking for the CD3 marker?
Yes.
Oh, okay.
Okay. So that is about cluster differentiation. And few points about complement system. I told you the complement activation. One of the mechanisms of activation of antibody is by activating the complement system. Okay, so what is this complement system? Complement system is named as they complemented the effects of antibodies. Okay, and these complements are polypeptides or proteins acting as enzymes, proenzymes or binding proteins. Okay, so there are several proteins, especially they are designated C1 to C9 proteins. Complement proteins are named C1 to C9. Okay, and these proteins can act as enzymes, proenzymes or binding proteins. And most of them are formed in the liver. Okay, so that is complement system. And how this complement system is activated? I told you there are two important pathways activated by antibodies. Isn't it? Classical pathway and alternate pathway. Actually, there are 3 pathways for activation. Of this, 2 pathways are activated by antibodies. Okay, and this classical pathway is triggered by, especially by the immune complexes. That is, immune complex means antigen antibody complex. Okay, antigen antibody complex. And
Ma'am, could you please show the previous slide once?
yes. Okay.
Thank you, ma'am.
And 3 pathways are there: classical pathway, alternate pathway, and Mannose binding lectin pathway. You just know the names of these 3 pathways. This is from, as this is given in your GANO, I am mentioning this. Okay, the detailed complement pathway is important in microbiology. Okay, but now you just know that what is this complement system and what are the different pathways. There are 3 pathways: classical pathway, alternate pathway, and Mannose binding lectin pathway. That much is important for you. And what are the effects of this complement pathway? Shall I change the slide? Just take down the names of the pathway only.
മാം ഇത് മുന്നേ പറഞ്ഞ ആ പാത്വേ
Yes, same thing. Classical pathway and alternate pathway ആണ് immunoglobulins activate ചെയ്യുന്നു പറഞ്ഞത്. There is one more pathway called Mannose binding lectin pathway. അത് എന്താണെന്ന് you will not understand. That is why I am not going to that pathway. In that pathway, a lectin protein will bind to the Mannose component of the antigen, and that will cause the activation of complement. That is the 3rd pathway. That is not related to antibodies, immunoglobulins. Okay. Okay, shall I change?
Yes, ma'am.
Yes, ma'am.
Okay, lot of portions to be done. Okay, and then what are the effects of these complements? They can, they will help kill the invading organism by the opsonization, chemotaxis, and eventual lysis of the cell. In opsonization and chemotaxis, we have seen chemotactic factors, we have or opsonization. We have seen that. Yes, very good, very good. Complement proteins are involved. They are acting as the opsonizing agents. Okay, so they help kill invading organism by these 3 mechanisms. Then they serve as a bridge from innate to acquired immunity by activating the B cells and aiding immune memory. Okay, they can help the formation of memory cells. And they help dispose of waste products after apoptosis. Okay, in apoptosis also they have roles. So these are important effects of complement proteins and how they cause cell lysis. Cell lysis is brought about by inserting proteins. They will cause activation of certain proteins called perforins. And these perforins are inserted on the cell membranes, and these will create holes on the cell membrane. What will happen when there are, when holes are found on the cell membrane? They will permit free flow of ions from the cells. That will cause the disruption of the membrane polarity, and that will lead to cell lysis. Okay, so how these complements are causing cell lysis? They will activate certain proteins, or they will cause insertion of certain proteins called perforins into the cell membranes, and this will create holes which will permit free flow of ions and thus disruption of membrane polarity. And this is the same mechanism done by cytotoxic T cells and also the NK cells. Okay, so that is about complement proteins. And there are certain things, there are certain soluble components involved in immunity. That is, they are called cytokines. What are these cytokines? Cytokines are nothing but hormone-like substances that act in a paracrine fashion to regulate immune responses. Paracrine, endocrine, all those effects you will learn later. And so they are hormone-like substances that act in a paracrine fashion. Paracrine means locally. Okay, mainly local action. Also have, some of them have got systemic action, but mainly local action. Local means substance in the area where the substance is formed. Okay, and they will regulate immune responses. So when you are asked about the regulation of immune response, you should write about the cytokines. And they are secreted by lymphocytes, macrophages, endothelial cells, neurons, and glial cells. Okay, there are different classes of cytokines like interleukin, chemokines, like that. Okay, what is this interleukin? Interleukins are, the name came from their role in communication between leukocytes, interleukin. Okay, this is shortly written as IL. Okay, interleukin, there are different numbers of interleukins. They are named in after the numbers. Okay, like interleukin 1, interleukin 2, like that. And they have systemic as well as local paracrine effects. Like some of the interleukins like interleukin 1, 6 and TNF alpha, tumor necrosis factor alpha, cause fever. And interleukin 1 increases slow wave sleep and reduces appetite. All these are part of inflammation. Okay, when you have inflammation, you will feel reduced appetite, severe inflammations. All these are because of the systemic effects of interleukins. Okay, and substances called chemokines. They have main role in chemotaxis. That is, substances that attract neutrophils and other WBCs to sites of inflammation or immune response. Okay, and they play role in regulation of cell growth and angiogenesis. We have discussed certain factors along with the regulation of leukopoiesis, isn't it? Certain growth factors and certain interleukins are involved in the regulation of leukopoiesis. Interleukin 3.
Yes.
Yes, involved in almost all the stimulates the growth of almost all the cell lines, and they that is why the interleukin 3 is known as multi-CSF, colony stimulating factor, like that, isn't it? Colony stimulating factors, all those things are coming under cytokines. Okay, so that is about the components of immune system. Next, we will see the mechanisms involved in the different immunity, like innate. Yes.
Slow wave sleep and
that you will learn in sleep. Slow wave, you have, sleep, you have two stages of sleep: slow wave sleep and rapid sleep, rapid eye movement sleep. Okay, rapid eye movement and non-rapid eye movement sleep. NREM and REM sleep and the different stages are there that you will learn in sleep in CNS, nervous system. Okay.
Okay, ma'am.
Okay. Okay, so these are the components involved in immune system. Next is about the mechanism of innate immunity. We have seen the important components involved in innate immunity. They are mainly the barriers, isn't it? And the innate immunity components.
Mechanical barriers.
Mechanical
like
barriers.
Physiological
barriers.
Yes.
Physiological barriers.
NK cells.
Yes, NK cells. And how
Macrophages.
how these components are causing the innate immunity. So that is about the mechanism of innate immunity. Okay. And this is the ancient mechanism, or it is called first line of defense against infections. Okay, this is the point given in your Ganong textbooks. So first line of defense in Ganong, it is given as innate immunity. Okay, innate immunity is the first line of defense. Okay, and it is triggered by receptors that bind sequences of sugars, lipids, amino acids, or nucleic acids that are common in bacteria, fungi, etc. Okay, so how this innate immunity or these barriers or these cells are detecting the antigens? എങ്ങനെയാണ് ഒരു foreign antigen ഇത് detect ചെയ്യുന്നത്? They are identifying certain patterns on the bacteria or fungi. Okay. So these cells like NK cells or the barrier cell, epithelial cells or the macrophages can also form part of innate immunity. Okay, and these cells will recognize certain patterns on these bacteria or fungi, and that will activate them, or they have certain receptors, or these cells will have certain receptors that can bind to these particular patterns on the bacteria or fungi that will cause activation of these cells, and that will lead to certain events inside the cells and finally causing the destruction of the antigen. So there are, which are the important cells involved in innate immunity? They are the phagocytes, or the phagocytes which can be neutrophils or macrophages, then dendritic cells, and many other cells such as epithelial cells. They can act as the antigen presenting cells, or the they can respond to these antigens because they express receptors on their surface. Receptors on their surface that sense the presence of infectious agents and substances released from dead cells. Okay, മനസ്സിലായോ? എങ്ങനെയാണ് അതിന്റെ മെക്കാനിസം എന്നാണ് പറയുന്നത്. These cells will have certain receptors that can sense the presence of infectious agents and substances from dead cells. Okay. And this receptor protein was first identified in a fruit fly called Drosophila. You might have heard.
Drosophila melanogaster.
Melanogaster.
Entrance question ആയിരുന്നില്ലേ? And so innate immunity was first studied in this fruit fly called Drosophila, and they identified a receptor protein present on the in the Drosophila called, that protein was named as TOL. That is called TOL protein. Receptor protein in Drosophila called TOL protein. Okay, so these receptors are called TOL receptors. And in humans also, they found that humans and vertebrates, there are certain receptors similar to this TOL protein called, they are called TOL-like receptors. Okay, so these TOL-like receptors are the receptors involved in innate immunity. മനസ്സിലായോ? Okay, and which are the TOL-like receptors? How these TOL-like receptors identify these infectious agents? These TOL-like receptors, there are a lot of TOL-like receptors. They are called as pattern recognition receptors because they recognize certain patterns on the bacteria or fungi or these infectious agents or on the dead cells. And that is why they are called PRR, pattern recognition receptors. And some examples of these receptors are TLR4. All these are given in your Ganong. That is why I'm stressing these points, because this can be asked as one-word questions. Okay. So TOL-like receptors, TLR4 can bind bacterial lipopolysaccharides. TLR2 can recognize lipoproteins. TLR6 along with TLR2 can identify peptidoglycan. Okay, so all these receptors can identify these particular patterns. TLR5 recognizes flagellin molecule. TLR9 recognizes bacterial DNA. Like that. Only few examples you should know. Okay. Just know that they are pattern recognition receptors. That is, they are called TOL-like receptors. Okay, and there are several other receptors, which are, these TOL-like receptors are mainly present on the cell membrane, and there are certain other receptors which are present in the cytoplasm. They are called intracellular PRR, pattern recognition receptors, like a protein called NOD protein. This is important because mutation in gene for NOD2 is causing a condition called Crohn's disease. Okay, that can be asked as a one-word question. അതുകൊണ്ട് മാത്രമാണ് ഞാൻ ഇത് പറയുന്നത്, because I know it is difficult for you, but it is given in your Gano. That is why I'm mentioning this point. Okay, intracellular pattern recognition receptors, example NOD protein. And mutation in gene for NOD2 can cause Crohn's disease. And there are certain other pattern recognition receptors like C-type lectin receptors and RIG-like receptors. Just know the names. This is just a diagram showing these 4 types of receptors, which are acting as the pattern recognition receptors. Okay. Just ഈ പേര് മാത്രം അറിഞ്ഞാൽ മതി കേട്ടോ. Okay, and what will happen when these receptors will bind to these certain patterns on these bacteria or fungi? What will happen? These receptors in turn activate various defense mechanisms like release of cytokines, activation of phagocytes, then production of antibacterial peptides, activation of complement system, and several proteolytic cascades. All these are the effects produced by the stimulation of these receptors. Okay, they can also, these intracellular events can activate transcription of genes for a variety of proteins involved in immune responses. Several proteins are formed which can act as these antibacterial peptides, or they can help in the lysis or killing of these infectious agents. Okay, so this is the mechanism of innate immunity. കിട്ടിയോ? മനസ്സിലായോ?
Yes,
എന്തെങ്കിലും മനസ്സിലായോ?
Yes, ma'am.
കുറച്ച് ടഫ് ആയിട്ടുള്ള പോർഷൻ ആണ്. ഓക്കെ, ടീസലിനെ കൂടി പറയണമെന്നുണ്ടായിരുന്നു, പറ്റുമെന്ന് തോന്നുന്നില്ല. Okay, so this is the innate immunity. So we have discussed today, we have discussed what is MHC protein, what is the what are antibodies, and about the structure and different classes of antibodies, and complement system, cluster differentiation, cytokines, isn't it? And after that we discuss the mechanism involved in innate immunity. In the next class we will see the mechanisms of adaptive or acquired immunity of this. First we will see the T cells, or the role of T cells in immunity, or the cell mediated immunity first, and after that humoral immunity. Okay, and for that you should just read the topic and come. Role of T lymphocytes in immunity, you can read your textbooks and come. Okay, then only you will understand this. അതിൽ ആദ്യത്തെ കുറച്ച് കാര്യങ്ങളൊക്കെ repetition ആണ്, types of T cells, and one important point is about T cell receptor. T cell receptor, we will see there are certain receptors present on the T cells that we will discuss briefly about the structure of T cell receptor and the mechanism of activation of T cells. Okay.
Yes, ma'am.
Okay, ma'am.
ഓക്കെ, എന്തെങ്കിലും ഡൗട്ട് ഇന്ന് ചോദിക്കാനുണ്ടോ, അല്ലെങ്കിൽ നെക്സ്റ്റ് ക്ലാസ് കൂടി കഴിഞ്ഞിട്ട് ചോദിച്ചാൽ മതിയെങ്കിൽ വി ക്യാൻ ആസ്ക്. ഇന്നത്തെ ക്ലാസ്സിൽ എന്തെങ്കിലും ഡൗട്ട് ഉണ്ടോ? എല്ലാരും പോയോ? Any doubts? Yes. എല്ലാവരും ഉറങ്ങിപ്പോയോ?
Hello, ma'am. We can hear you, ma'am.
ഇല്ല മാം.
ഉറങ്ങിയിട്ടില്ല മാം.
ഉറങ്ങിയിട്ടില്ല. ഓക്കെ, ഡൗട്ട്സ് ഉണ്ടോ? ഇല്ലെങ്കിൽ നമുക്ക് ലീവ് ചെയ്യാം.
Next class ഉണ്ട് ma'am.
Okay, we will stop here.
Yes, ma'am.
Thank you, 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 ma'am.
Ma'am.
Thank you, ma'am.
Everyone, please mute.
Pauli, please mute.
Good morning, students.
Good afternoon, sir.
ഗുഡ് മോർണിംഗ് യാസിൻ.
എല്ലാവരും ഉറങ്ങുകയാണോ? പഠിക്കാൻ
ഗുഡ് മോർണിംഗ് അല്ലേ? ഗുഡ് ആഫ്റ്റർനൂൺ. സെബ്
ഞാൻ ഓർത്തു ഫഹദ് സാർ വന്നേക്കും ഒന്നുകൂടെ പറയാൻ.
Yes, ma'am.
Okay, I'm sorry for the delay. There was some kind of issue there, like with my Zoom, so I couldn't actually log in. So your class stopped at 10:05, no? Kavita's class?
Yes, ma'am.
Yes, ma'am.
Yeah, you are having class from morning, I mean from 8 o'clock onwards?
Yes, ma'am.
Okay, so now you had more than enough break, no?
Yes, ma'am.
Okay, so shall we continue with the class?
Yes, ma'am.
Okay.
Yes, ma'am.
So let us not waste the time and So what did we study in the previous class?
Primary
Primary
hemostasis and
hemostasis.
secondary hemostasis.
Yes, in the previous class we have started with hemostasis. So I have told you what are the different steps of primary hemostasis, and we have started with the steps of secondary hemostasis, right? So can anyone please tell me what are the different steps of primary hemostasis? Yazin? Yazin?
Yes,
Yazin?
ma'am.
Yes, Yazin, tell me.
Vascular spasm.
Yes,
platelet-
vasospasm or vasoconstriction. Vasoconstriction, yeah.
Yes. Then plaque formation and
Platelet, yes, platelet plaque formation.
and blood coagulation.
Coagulation, then who's laughing there?
ma'am.
Kainyo. Can anyone please tell me what are the different steps of hemostasis?
Primary hemostasis.
Yeah.
Secondary clot retraction.
I have told you very specifically. I have told you, in the recorded classes also, I have very specifically told you that you have to elaborate upon all these steps when the question comes like, what are the steps of hemostasis? If the question is specific like primary or secondary, you can write like that. But if the question is like, what are the steps of hemostasis, you have to specifically explain all these steps. I have very clearly stated that. You see, you just understand that we are taking so much of pain for taking all these classes, for recording and again taking the live session is like very, very hectic. No? Why? Because we are doing all this is for your sake. No, it's not for our sake. It's for your sake. Okay, so you should at least show that courtesy to, at least read and come. Hope it's clear.
Yes, ma'am.
There is nothing funny about that. Okay, so we'll continue with the remaining portions of hemostasis. So I have briefly told you what are the steps of secondary hemostasis. Somebody's phone is mute. Please, for all of you, mute your devices. All of you mute your devices. I'll start sharing my screen right now. Yeah, 1 second. I'm audible now. Previously, my PGs told me that in the previous session there were some kind of clarity issues. So, like, is it clear now?
Yes, ma'am.
Yes,
ma'am.
Okay. Okay. So next is with the hemostasis. So in today's session, we are actually going to explain what are the different mechanisms of intrinsic and the extrinsic pathway of coagulation, and I will discuss the natural anticoagulant mechanisms, or why blood doesn't clot. Okay, and we'll see how each mechanism acts. Are my slides visible to you? Are my slides visible to you?
Yes, ma'am.
So I have told you that when a blood vessel is injured, the first thing that happens is primary hemostasis, and the primary hemostasis can control bleeding to some extent, but if the injury is of sufficient size, then the platelet plug won't be sufficient to seal the vascular injury. Then the secondary hemostasis will come into action. So where you have in the blood, you have a lot of procoagulant substances, like more than 50 procoagulant and anticoagulant substances are there. But in a normal circulation, you have more of anticoagulant substances than the procoagulant substances are in. Procoagulant substances are in inactive form. But when this blood vessel is injured, these mechanisms will get activated, and if the tissue injury is of sufficient size, then the mechanism can act within seconds, within a few seconds, whereas if it is of small size, then it will take some minutes. Whatever it is, and these clotting mechanisms will get activated, and you have 12 clotting factors. So what are the names of the clotting factors? What is factor 2? What is factor 2? Prothrombin. What is factor 10?
Stuart factor.
Stuart factor. Okay. What is factor 13?
Fibrin
stabilizing factor. So I have to. Okay. So I have told you these 12 clotting factors are the most important. Along with this, you have some other substances like high molecular weight, kininogen, prekallikrein, kallikrein, phospholipid, etc. So now I'll go into the pathway. The pathway exact pathway. So we have discussed the 3 basic steps involved in secondary hemostasis. What are they? 3 basic steps involved in
Activation of
Formation of prothrombin
yes, formation of prothrombin activator, conversion of prothrombin to
Thrombin activator.
Conversion of soluble
thrombin convert
fibrinogen to fibrin.
ഫിബ്രിൻ. ഓക്കെ, അപ്പൊ ഇനി നമുക്ക് നോക്കാം എങ്ങനെയാണ് ഈ പാത്വേ ആക്ട് ചെയ്യുന്നതെന്നും എങ്ങനെയാണ് ഫൈനലി ഈ ഫിബ്രിനോജൻ ഉണ്ടാവുന്നതെന്നും നമുക്ക് നോക്കാം. സോ ഐ ടോൾഡ് യു there are how many pathways are there? You have 2 pathways. ഈ പറയുന്ന ഓരോ കാര്യവും എക്സ്ട്രീംലി ഇമ്പോർട്ടന്റ് ആണ് കേട്ടോ, ഭയങ്കര ഇമ്പോർട്ടന്റ് ആണ്. ഇതെല്ലാം A to Z നിങ്ങൾ അറിഞ്ഞിരിക്കണം കേട്ടോ. So I will discuss both the pathways of coagulation. First I will deal with the intrinsic pathway of coagulation. Okay, so this is also called as the contact pathway. Why it is called as a contact pathway? Because this mechanism is initiated by the contact of the factor 12 with the electronegative surfaces. So this is activated by contact. That is this in vivo activation of this pathway. ബോഡിക്കുള്ളിൽ ഈ പാത്വേ എങ്ങനെയാണ് ആക്ടിവേറ്റഡ് ആവുന്നത്? ഇറ്റ് ഈസ് ആക്ടിവേറ്റഡ് ബൈ ട്രോമ ടു ബ്ലഡ് ഓർ ഇറ്റ് ഈസ് ബൈ ദ കോൺടാക്ട് ഓഫ് ദ ബ്ലഡ് വിത്ത് ദ സബെൻഡോത്തീലിയൽ കൊളാജൻ. Okay, and this pathway can also be activated in vitro. How? When this blood comes in contact with electronegatively charged vegetable surface. For example, when the blood comes in contact with glass. Clear? When blood comes in contact with glass being an electronegative surface, it can initiate intrinsic pathway. Clear? Yes. Okay, so we will look at the pathway. Okay, so the intrinsic system, you can see here this factor 12 when it comes in contact. Can you see here? One second. Okay, so can you see here? This is the factor 12. When it comes in contact with negative surface, what will happen to electronegative when it comes in? Yes?
Your audio is breaking.
No, I was not talking anything. Is that visible now? I mean, is that audible now?
Yes, ma'am.
Okay, I was not talking anything in between. Okay, so this is factor 12A. This 12A along with HMW kininogen. What is that? HMW?
High molecular weight.
High molecular weight kininogen. And there is another substance called as kallikrein. In the presence of this substance, this factor 12A will cause the conversion of factor 11 to 11A. Is that clear? Is that step clear? Okay. So what will happen to this factor 11A? The 11A will go and it will cause the conversion of factor 9 to 9A. And the 9A will cause the conversion of 10 to 10A. But you can see here, this is a very, very important step. You look at here. You focus on this area. You focus here. So along with factor 9A, you can see that you have not only factor 9A, you have the activated factor 8A. Can you see here? You have the activated factor 8A along with calcium and phospholipid. So how was this factor 8A formed? Factor 8A, from where is it being formed? Any idea?
From factor 8.
From factor 8.
No. From how does it get activated?
One will be
Endo?
factor.
Von Willebrand factor is not activating it. Von Willebrand factor is it's a transporter of factor VIII.
Carrier for.
Yes.
By calcium.
Not exactly. Calcium has a role, but mainly which substance or which clotting factor is responsible for its action?
Phospholipid.
Yes, very good. Thrombin. Right? Thrombin. Appa ningal vicharikum thrombin undavan ponalle ullu ennu. So whenever there is this activation, I have told you this platelet granules contain a lot of substances. No, thrombin is also one constituent present there. So thrombin, some minor amount of thrombin will be formed there. So that thrombin will cause activation of factor 8 to 8A and factor 5 to 5A. അപ്പൊ അതിന്റെ പ്രത്യേകത എന്താന്ന് വെച്ച് കഴിഞ്ഞാല് വളരെ കുറഞ്ഞ അളവിൽ തന്നെ ഈ സാധനങ്ങൾ ഉണ്ടായി കഴിയുമ്പോൾ they will cause further activation and formation of all these substances. Is that clear? Is that clear? Factor 8A and factor 5A here. These are actually formed or they are stimulated by the very minute amounts of thrombin formed in the circulation. So this factor 9 to 9A, this 9A along with activated factor 8A, phospholipids. From where do you get this phospholipid? From where do you get this phospholipid?
Inner membrane of platelets.
Yes, which part of the membrane? Is it the outer layer or the inner layer?
Inner layer.
Inner layer. Okay,
Proteins.
so this, yes, very good. So this phospholipid, I'm sorry, calcium activated factor 8A together with 9A causes the conversion of factor 10 to 10A. Can you see here? It causes the conversion of 10 to 10A. So this 10 to 10A along with factor 5 to 5A, phospholipid and calcium, it acts as the prothrombin activator. Clear? It acts as the prothrombin activator, which causes the conversion of prothrombin to thrombin. Clear? Clear?
Yes, ma'am.
Okay. I'll repeat this pathway once again. We have been discussing about the intrinsic system. So the factor 12 will get activated to 12A when it comes in contact with electronegative surfaces and the presence of high molecular weight kininogen and kallikrein. This factor 12A causes conversion of 11 to 11A in the presence of HMW kininogen. 11A causes conversion of 9 to 9A, and the 9A in the presence of activated factor 8A, phospholipid and calcium, causes the conversion of 10 to 10A, and the 10A in the presence of phospholipid, calcium and activated factor 5A, it acts as the prothrombin activator and it causes the conversion of prothrombin to thrombin. And this thrombin which is being formed has got many actions. Okay, this thrombin actually causes the conversion of fibrinogen to fibrin, and this thrombin will also cause, this step is also very important. It will also cause the conversion of factor 13 to 13A. So what is factor 13A? What is factor 13A?
Fibrin
stabilizing factor. So what this fibrin stabilizing factor will do? It causes stabilization. This entire fibrin threads in a stable form like what is the role? I mean, that is the role of fibrin stabilizing factor. Clear? ഇത്രയും പറഞ്ഞത് മനസ്സിലായോ? ഈ പറഞ്ഞ അത്രയും മനസ്സിലായോ?
Yes, ma'am.
Okay. Now we will look into the extrinsic system. Extrinsic system. So how is this extrinsic system activated? How is this extrinsic system activated?
Tissue factor.
Yes, it is activated by injury to the tissue. So this tissue, when it gets injured, it causes the release of a substance called as tissue thromboplastin. What is that TPL? It is Tissue.
thromboplastin.
Which factor is that? Which factor is that?
Factor 3.
Factor 3. Very good. It is factor 3. So this substance which is formed, it is a complex formed from tissue released from the site of injury along with the phospholipid complex. It causes the activation of factor 7 to 7A. What is 7 called as? Factor 7 called as? Pro-converting. Okay. And this factor 7A has got two actions. It causes the activation of factor 9A and also it causes the activation of factor 10A, ultimately resulting in the formation of the prothrombin activator. But when the amount of factor 10A becomes more than what is needed, it causes activation of another substance called as tissue factor inhibitor. What does it form? It causes tissue factor inhibitor. That forms a complex with this TPL, this phospholipid and calcium, and it causes inhibition of factor 7A when sufficient amount of factor 10A is being formed. ഇത്രയും കാര്യങ്ങൾ പറഞ്ഞതിനകത്ത് എന്തെങ്കിലും സംശയമുണ്ടോ? രണ്ട് പാത്വേയും അതിന്റെ ഈച്ച് ആൻഡ് എവറി സ്റ്റെപ്പ് നിങ്ങൾ കൃത്യമായിട്ട് എഴുതാൻ നിങ്ങൾക്ക് അറിയണം. അപ്പൊ ഈ പറഞ്ഞിരിക്കുന്ന all these short forms should be expanded. The expansion of those short forms should be written on the corner of the paper. കേട്ടോ? അതിന്റെ എക്സ്പാൻഷൻ സൈഡിൽ കോർണറിലായിട്ട് എഴുതിയിട്ടുണ്ടാവണം. മനസ്സിലായോ പറഞ്ഞത്? ഈ പറഞ്ഞത് അത്ര മനസ്സിലായി? ഓക്കെ. അപ്പൊ ഞാനൊരു കാര്യം ചോദിക്കട്ടെ, എന്തിനാണ് നമുക്ക് രണ്ട് പാത്വേകൾ? ഏതെങ്കിലും ഒരു പാത്വേ പോരായിരുന്നോ?
ഇന്റേണൽ ബ്ലീഡിംഗ് ഉണ്ടെങ്കിൽ അറിയാനായിരിക്കും. ഇന്റേണൽ ബ്ലീഡിംഗ് സംഭവിച്ചാൽ
ഇന്റേണൽ ബ്ലീഡിംഗ് മീൻസ് വാട്ട്?
ടിഷ്യൂനൊക്കെ ഡാമേജ് വന്നാൽ.
ടിഷ്യൂന് ഡാമേജ് വരാതെ ഒരിക്കലും ബ്ലീഡിംഗ് ഉണ്ടാവുമോ? ടിഷ്യൂന് ഡാമേജ് വരാതെ എങ്ങനെയാ ബ്ലീഡിംഗ് ഉണ്ടാവണേ?
Ma'am, both the systems will help each other.
Yes, how do they help each other?
Blood കൂടെ ആണെങ്കിൽ
Ma'am, it's not always required that
ഒന്ന് വർക്ക് ചെയ്തില്ലെങ്കിൽ വേറൊന്ന് വർക്ക് ചെയ്യണം.
No, I will tell you. Have you heard of hemophilia? What is the problem in hemophilia?
Yes,
You
ma'am.
have? Yes, what is the problem there?
Continuous loss of
Loss of coagulation factor 8, 9 and 10 causes hemophilia factor 8, 9 and 11 causes hemophilia A, B and C. Okay, so my question is, if it is hemophilia A, which coagulation factor is affected?
Anti-hemophilic factor 8.
Factor 8. So which pathway will be affected mainly? Intrinsic pathway. So you don't require the extrinsic the factor 8A illengalum factor 7A ki 10 to 10A akite it can form prothrombin to thrombin alle. So why does bleeding occurs there? Hemophilia A patients എങ്ങനെയാ present ചെയ്യുന്നത്? They present with bleeding, right? So why bleeding occurs there? You have both the pathways. Only one pathway is affected, the other pathway is intact. So why bleeding occurs there?
രണ്ടും ഒരുപോലെ നടക്കണം.
കോംപ്ലിമെന്റ് ചെയ്താ നടക്കും.
എങ്ങനെയാ കോംപ്ലിമെന്റ് ചെയ്യുന്നത്? How do they complement each other?
I said because like in all those patients who have got hemophilia, the calcium count is less. Therefore, like it can't help in the process like of intrinsic.
No, calcium is one second. Hello. ഞാൻ കുറച്ച് കഴിഞ്ഞിട്ട് വിളിച്ചാൽ മതിയോ? ഹലോ. ഹലോ. എന്തോ?
Enough clot is not formed.
Why the clot is not being formed? The extrinsic pathway is intact, no? Extrinsic
pathway factors
endo calcium has nothing to do with, calcium is intact. How does calcium get affected in all these things? നിങ്ങൾ ഒരു കാര്യം മനസ്സിലാക്കണം, കാൽസ്യം കുറഞ്ഞിട്ട് ഒരിക്കലും ബ്ലീഡിംഗ് ഡിസോർഡർ ആയിട്ട് മാനിഫെസ്റ്റ് ചെയ്യത്തില്ല. When the calcium count is like if it reaches around 5 or 6 milligram percentage, the first thing that will manifest is increased neuromuscular spasm. The patient will die due to neuromuscular hyperexcitability. Bleeding ഉണ്ടാവണമെന്നുണ്ടെങ്കിൽ അതിലും കുറഞ്ഞാൽ മാത്രമേ അതിലും കുറഞ്ഞ ലെവലിൽ എത്തിയാൽ മാത്രമേ ബ്ലീഡിംഗ് ഉണ്ടാകത്തുള്ളൂ. അപ്പൊ ക്ലിനിക്കലി ബ്ലീഡിംഗ് മാനിഫെസ്റ്റ് ചെയ്യുന്നതിന് മുൻപ് തന്നെ calcium has no role in the cause of bleeding here.
Vitamin K.
സിസ്റ്റത്തിലും ഫാക്ടർ 8A വേണ്ട.
Factor A, എൻ്റെ question ഇത്രേ ഉള്ളൂ. Factor A is affected, only the intrinsic system is affected, the extrinsic system is intact. So why the bleeding occurs there?
When the clot is formed, but it is not very much.
8A ഉണ്ടല്ലോ.
Okay, so shall I tell you the answer?
Yes, ma'am.
Yes, ma'am.
Okay, so this intrinsic system, as somebody have rightly told, they actually complement each other. Okay, so what the extrinsic system will do is, extrinsic system is actually an explosive pathway. Once the tissue is injured, within a few seconds, the extrinsic pathway will get activated. Whenever the tissue injure, tissue thromboplastin will be released, immediately the extrinsic pathway will get activated. Suddenly it will cause the activation of the coagulation pathway, and thrombin that finally that causes activation of factor 8A and 5A, and this intrinsic system is actually very slow to act, but it is more long-lived. It is long-lived, but very slow to start with. Whereas extrinsic system is very rapid to act with, but it is a short-lived system. So thus they actually complement each other. So in hemophilia, even if there is actually the intact extrinsic system, this and extrinsic being short-lived, it cannot produce a clot of sufficient size. You require both the systems to be intact to produce a normal coagulation. Is that point clear for all of you?
Yes, ma'am.
A concept clear, Anna?
Yes, ma'am.
Yes, ma'am.
Okay. So this pathway, that is from this prothrombin activator formation, you call this pathway as the common pathway. So you can see here the prothrombin activator causing. So this is the junction of the intrinsic and the extrinsic. Why there is somebody's phone is not mute? Please mute your device. Yes, please, all of you mute your device. Yes, please mute your device. Shamila. Okay, so that is the common pathway, that is the factor 10A along with the factor 5A, phospholipid and calcium, causing the activation of prothrombin to thrombin, and thrombin causing the conversion of fibrinogen to fibrin, as well as fibrin stabilization, constitutes the common pathway. Okay, and the thrombin, it causes conversion of insoluble fibrinogen to fibrin. So what is this? So that we will discuss. Oh, sorry. Okay, so these are the summary of the steps of coagulation: formation of prothrombin activator, conversion of prothrombin to thrombin, and conversion of fibrinogen to fibrin. See, these are the basic steps in coagulation. Okay, so first is the formation of prothrombin activator. So normally blood doesn't clot in circulation. Why? Because the clotting factors are in inactive form, and the blood contains also some amount of anticoagulants. Okay, so prothrombin activator is formed by a cascade of reaction in which activation of one leads to activation of another. I have told you it is like a chain reaction. When one factor gets activated, it will act as an enzyme which causes the next series of coagulation factors to get activated. So they act both as a substrate and as an enzyme. And the prothrombin activator is formed by different pathways, that is the intrinsic and the extrinsic pathway. But even though those two pathways, they help in coagulation, they actually complement each other. Intrinsic pathway is actually slow to act, but it is a long-lived system, whereas extrinsic pathway is an explosive, immediately acting mechanism, but it is a short-lived mechanism. And the intrinsic system, it is actually initiated by activation of factor 12, and extrinsic system is activated by release of tissue thromboplastin. So all these things we have discussed. Alle nammal ini veendum veendum parayanda karyillalo undo okay. So next is the conversion of prothrombin to prothrombin. So this prothrombin, it's a very, very important coagulation factor. Actually, this prothrombin it acts as both as a procoagulant as well as an anticoagulant substance. Okay, it has got both procoagulant activity as well as anticoagulant activity. Right? The prothrombin, it's a plasma protein which is synthesized by factor liver in the presence of vitamin K. I will tell you in detail about this later. So this is a vitamin K dependent coagulation factor. Which are the vitamin K dependent coagulation factors? Which are the vitamin K dependent? 2,
Factor 2, 7, 9, 10.
Excellent. 2, 7, 9, 10. So even very, that will accelerate the coagulation process by activating factor 5, 8, 13, etc. Manasilavunundo? Is that clear? Okay. So next is the conversion of fibrinogen to fibrin. So fibrinogen is actually, it is a soluble protein which is synthesized by the liver. So what this thrombin will do? Thrombin, I'm sorry. This thrombin, it will remove 4 polypeptides from each molecule of fibrinogen. Oro fibrinogen monomer nathonum adu 4 polypeptide ne angotu remove cheyidu maattum. Annittu endu cheyum ee baaki vanna saadhanangalellam kude they will attach with each other. So that will result in the formation of a fibrin thread. So this fibrin thread will, the fibrin monomers, they will polymerize to form the fibrin thread. Okay. This fibrin is initially, it is a loose mesh of interlacing strands. What the factor 13 will do? It will form cross linkages between the fibrin threads, and they will cause the stabilization of the fibrin fibers. Okay, and this dense meshwork. So you can see here, look at this picture. You can see here, you have a lot of trapped blood cells within this meshwork of the fibrin thread. Clear? So factor 2 causes mainly fibrin thread formation, whereas the in between the fibrin thread, that connection is strengthened by factor in between connections of fibrin thread are strengthened by which factor?
Factor 13.
Factor 13. Fibrin stabilizing factor. Stabilizing factor. So this diagram is taken from Guyton. So this is in a nutshell of what we have discussed in secondary hemostasis. So prothrombin just converted to prothrombin by prothrombin activator. Thrombin has got several actions. This thrombin causes conversion of fibrinogen to fibrin monomer and fibrin fibers, which is stabilized by this fibrin stabilizing factor, and this fibrin stabilizing factor is activated by which factor? Fibrin stabilizing factor is activated by thrombin. So this fibrin fibers, they will develop cross linkages in between fibrin fibers. So that is in nutshell regarding the coagulation process. So is that process, whole process, clear for all of you? എന്തെങ്കിലും സംശയമുണ്ടോ ഈ പറഞ്ഞ ഇത്രയും കാര്യങ്ങൾക്കകത്ത്? ഇത്രയും പറഞ്ഞതിനകത്ത് എന്തെങ്കിലും സംശയമുണ്ടോ?
No, ma'am.
Okay. Is my screen visible?
Yes, ma'am.
Yes, ma'am.
Okay. So we have discussed 3 important steps of hemostasis. Which are they? Which are they? Primary hemostasis. Primary hemostasis, which basically involves primary hemostasis involving two major mechanisms: vasoconstriction and platelet formation. Then we have discussed about the secondary hemostasis, which basically involves the intrinsic and extrinsic pathway resulting in the formation of the fibrin threads. Next, what happens? നിങ്ങൾ ചെറുപ്പത്തിൽ കണ്ടിട്ടുണ്ടോ അറിയില്ല, നമ്മളെ മറിഞ്ഞു വീണിട്ട് നമ്മുടെ കാലിന്റെ മുട്ടൊക്കെ പൊട്ടി കഴിയുമ്പോഴത്തേക്ക് കുറച്ചു നേരം കഴിയുമ്പോൾ മുട്ടിലെ ആ ഇതെല്ലാം ഉണങ്ങി കഴിയുമ്പോൾ സ്കാബ് വന്നു കഴിയുമ്പോഴത്തേക്ക് what will happen? Have you ever seen? Have you ever noticed that after a fall or an injury, after some time, no, after a few hours or some, a scab will form at the site of injury, and you'll be able to see a very clear yellow fluid oozing from the site of injury. Have you ever noticed?
Yes, ma'am.
Yes, ma'am.
അല്ലേ, നമ്മുടെ ഒക്കെ ചെറുതിലെ മുട്ട് പൊട്ടിയിട്ട് അങ്ങനെ സംഭവിച്ചിട്ടില്ലേ നമുക്ക്?
അത് പഴുപ്പല്ലേ മാം?
പഴുപ്പല്ല, പഴുപ്പ് വരാൻ കുറച്ച് ദിവസം കൊണ്ട് എടുക്കും കേട്ടോ. പഴുക്കാൻ കുറച്ച് ദിവസം കൊണ്ട് എടുക്കും. മുറിവ് പറ്റുന്ന അന്ന് തന്നെ കാലിൽ ഇങ്ങനെ മഞ്ഞ കളറിൽ ഇങ്ങനെ വെള്ളം പോലെ വരും. ഇങ്ങനെ ഉണങ്ങി കുറച്ച് കഴിയുമ്പോൾ അതിങ്ങനെ ഉണങ്ങി പിടിച്ചിരിക്കും. കണ്ടിട്ടുണ്ടോ നിങ്ങള്? കണ്ടിട്ടില്ലെങ്കിൽ ഇനി മറിഞ്ഞു വീഴുമ്പോ നോക്കുക കേട്ടോ. സോ എങ്ങനെയാ അത് ഉണ്ടായ മഞ്ഞ വെള്ളം ഉണ്ടായത്? ഹൗ that fluid is being formed? And what is that fluid? What is that fluid?
Serum.
That fluid is actually your serum. What is the serum?
Blood without clotting.
Yes, very good. Blood without clotting factors. So why that fluid oozes? Why that fluid oozes?
Clot form ചെയ്തതിനു ശേഷം
ബാക്കിയുള്ളതല്ല actually. So this clot after its formation, after about 20 to 40 minutes and sometimes up to about 60 minutes or 1 hour, this clot will undergo retraction. Retraction എന്ന് പറഞ്ഞാൽ എന്തുവാ? What do you mean by retraction? Shrink or ചുരുങ്ങി പോണേനെയാണ് നമ്മൾ എന്ത് പറയണേ? Retraction. Okay, so why this clot retracts?
Because
why
of retracted protein. Retractile protein.
Yes, which are the contractile proteins present in the platelet?
Thrombostin,
actin and myosin. So these contractile proteins will make the platelet contract. So the fluid which has been entrapped within the clot will ooze out as serum. So what is serum? It is a yellowish fluid which is left after coagulation. So it is very similar to your plasma. What is plasma? What is plasma?
Fluid matrix of blood.
Yes, it is a fluid portion of your blood. Blood is a tissue, no? It's a fluid connective tissue. It has got a lot of cells and it has got fluid portion. So what is that fluid portion of blood called as? Plasma. What does plasma contain?
Water and
solids. Solids include organic and inorganic substances. Of the inorganic substances, the most important are the plasma proteins. Plasma proteins. How will you basically classify the plasma proteins?
Albumin.
Fibrinogen. So what is this serum? Is it plasma? Is it plasma?
Plasma without fibrinogen.
Plasma, it is almost similar to plasma. It is a fluid portion of the blood, but it doesn't contain the coagulation factors. So where are your coagulation factors?
They have been used up in clotted form.
Have all been used up during the coagulation process. Okay, so why is it called a serum? Serum? Why is it called a serum?
No clotting factors.
Serum?
No clotting factors.
Clotting factors.
എന്തുകൊണ്ടാ സീറം എന്ന് വിളിക്കുന്നേ?
ഗ്ലോബുലിനൊക്കെ ഉണ്ടായിരിക്കും.
ഗ്ലോബുലിൻ ഇല്ലാത്തോണ്ടാണോ? എന്തുള്ളതുണ്ട്?
യെല്ലോ കളർ ആയോണ്ട്.
യെല്ലോ കളർ ആയതിനെയൊക്കെ സീറം എന്നാണോ വിളിക്കുന്നേ?
ടിഷ്യൂ ഗ്രോത്തിന് സഹായിക്കുന്നു.
ടിഷ്യൂ ഗ്രോത്തിന് സഹായിക്കുന്നതുകൊണ്ട് സീറം എന്ന് വിളിക്കുവോ?
It's nutrition.
ഞാൻ പറയട്ടെ എന്താ വിളിക്കുന്നതെന്ന്.
Yes, ma'am.
Okay, ma'am.
High amount of serotonin. Why is it called as serum? It contains high amount of serotonin. എവിടുന്ന serotonin വന്നേ? Which is the source of serotonin?
Rates of breakdown of platelet.
Yes, it is from the breaking down of the granules of the platelets. Platelet granules. Is that clear? ഇപ്പൊ നിങ്ങൾക്ക് correlate ചെയ്യാൻ പറ്റുന്നുണ്ടോ? ഞാൻ ആദ്യം എന്തിനാ platelet function പഠിപ്പിച്ചെന്നൊക്കെ? Are you able to correlate all these things?
Yes, ma'am.
Okay. Okay. So what's the final step? So why so this clot retraction actually helps to make your vessel patent അല്ലെ? അല്ലെങ്കിൽ വസലിന്റെ അകത്ത് ഈ ക്ലോട്ട് ഇങ്ങനെ തടഞ്ഞിരിക്കും. പിന്നെ കുറച്ച് ദിവസം കഴിയുമ്പോൾ when this repair process and all is over, അവിടെ തന്നെ ക്ലോട്ട് ഇരുന്ന എങ്ങനെ ഇരിക്കും? Will the blood be able to flow normally? Will the blood be able to flow freely? No. So what should happen when its function is over? What should happen?
Clot is to be removed.
Clot should be removed. So how is the clot removed? By a process called as clot lysis, by a mechanism called as the fibrinolytic system. So the details of the fibrinolytic system I'll be discussing within a few minutes. But പരീക്ഷയ്ക്ക് ചോദിക്കുമ്പം നിങ്ങൾ അങ്ങനെ എഴുതരുത്, few minutes-നകത്ത് ഡിസ്ക്രൈബ് ചെയ്യുന്ന എഴുതരുത്. ഞാൻ ഇനി പറയാൻ പോണ കാര്യങ്ങൾ you have to write along with this fibrinolytic system. കേട്ടോ, ഡീറ്റെയിൽസ് ഞാൻ പറയാം. പറഞ്ഞത് മനസ്സിലാവുണ്ടോ? Are you able to follow the details of the fibrinolytic system? Soon I will be discussing. Okay. So you so now this is in yeah.
Ma'am, when like how it was before, when clot retraction takes place, ma'am, we had told only serum comes out, and serum is like the plasma protein, plasma without the clotting factors. But in fact, ma'am, isn't only the fibrinogen used? So what about the globulin as well as the albumin, ma'am?
Albumin and globulin doesn't have any role in. So your clotting factors are all actually a fraction of this. Only the fibrinogen is like factor 1. Other factors, they actually comes under the globulin class. So all the factors which are not used up in the coagulation process will be there in the serum.
Okay, ma'am. So isn't it right to tell, ma'am, serum along with the clotting factors except fibrinogen?
Yeah, not except fibrinogen. The other coagulation factors also will be used up, no?
Oh, okay, ma'am. Okay, yes, ma'am.
All the coagulation factors that will be used up during this process. So all those coagulation factors, I mean, this plasma excluding the coagulation factors are the, that is called as the serum. Is that concept clear?
Yes, ma'am. Yes, ma'am.
Because the coagulation factors will get used up during the coagulation process. Okay.
Ma'am.
Thank you, ma'am.
പ്ലാസ്മ തന്നെ, പ്ലാസ്മയിൽ ഉണ്ടായിരുന്ന coagulation factors മാത്രം നമ്മളെ coagulation നടന്നപ്പോൾ ഉപയോഗിച്ചു പോയി. അപ്പൊ അത് ഒഴിവാക്കിയിട്ടുള്ള ബാക്കിയുള്ള എല്ലാ സാധനങ്ങളും you call it as serum. പ്ലാസ്മ തന്നെയാണ്, പക്ഷെ except the coagulation factors. Clear?
Ma'am.
Oh, thank you, ma'am.
Okay.
ഇല്ല, സെറത്തിനകത്ത് സെറട്ടോണിന്റെ അളവ് കൂടത്തില്ലേ? അത് ഡെൻസ് ഗ്രാനുലിൽ അല്ലേ? ഡെൻസ് ഗ്രാനുലിൽ നിന്നല്ലേ വരുന്നത് ആ സെറട്ടോണിൻ അല്ലേ? അത് എങ്ങനെയാ വരുന്നത്? ഉള്ളിലല്ലേ ഡെൻസ് ഗ്രാനുല്?
അയ്യോ, all the platelet will get, ഈ ഇന്ന പ്രോസസ്സിന്റെ ഇടയ്ക്ക് ഒരുപാട് പ്ലേറ്റ്ലെറ്റ് പൊട്ടി പോകുന്നുണ്ട്. അങ്ങനെയല്ലേ നമ്മുടെ പ്ലേറ്റ്ലെറ്റിന്റെ ഉള്ളിലുള്ള ഫോസ്ഫോലൈപ്പിഡ് ഒക്കെ പുറത്തു വരുന്നത്. യു ഡോണ്ട് തിങ്ക് ദാറ്റ് ഓൾ ദ പ്ലേറ്റ്ലെറ്റ്സ് വിൽ റിമൈൻ ഇൻടാക്ട് ആൻഡ് എല്ലാ പ്ലേറ്റ്ലെറ്റും ഇങ്ങനെ ഭയങ്കര തടിയനായിട്ട് അവിടെ പോയിട്ട് ഇങ്ങനെ ഉരുണ്ട് നിൽക്കുകയാണെന്ന് വിചാരിക്കരുത്. ഈ പ്രോസസ്സിന്റെ ഇടയ്ക്ക് കുറെ എണ്ണമൊക്കെ പൊട്ടിപ്പോവും. അതിന്റെ നിങ്ങൾ സെക്കൻഡ് സ്റ്റെപ്പ് ഓഫ് പ്രൈമറി ഹീമോസ്റ്റേസിസ് ഏതായിരുന്നു? Which was the second step of primary hemostasis? What is happening there during platelet activation and secretion?
And secretion of granules.
There is secretion of. നമ്മൾ അവിടെ സ്റ്റെപ്പിനകത്ത് പറഞ്ഞില്ലേ ഗ്രാനുൾസ് എല്ലാം പുറത്തോട്ട് വരുന്ന? ഇല്ലേ, പറഞ്ഞില്ലായിരുന്നോ? നിങ്ങൾ ഇതെല്ലാം കോറിലേറ്റ് ചെയ്ത് പഠിക്കണേ, എന്നാലേ നിങ്ങൾക്ക് അത് കുറെ പ്രാവശ്യം വായിച്ചു നോക്കുമ്പോൾ you will be able to correlate all these things. അതിനെ ഞാൻ വീണ്ടും ഇതിങ്ങനെ പറഞ്ഞുകൊണ്ടേയിരിക്കണേ. Okay, so this diagram, actually the first one is taken from Guyton and the second one is from Genome. This is a summary of all these things. Namali parna hemostasis ella steps in dem oru summary aanide. When a blood vessel is injured, you can see the platelets will form a platelet plug here, then the fibrin thread forms, and finally the fibrin will form a strong mesh for fibrin clot forms, and finally the clot retraction occurs here. So this diagram is from Genome. You can see here vessel wall injury has happened here. What is 5-HT? 5-hydroxytryptamine is serotonin. Serotonin means serotonin is chemically 5-hydroxytryptamine. That is 5-HT. So you can see here the platelets will be recruited here, causes vasoconstriction as well as platelet plug formation. Along with that, the subendothelial collagen exposed to this thing causes activation of coagulation pathway. Thromboplastin also activates the coagulation pathway. Clotting mechanism will get activated, which will cause the activation of thrombin and causing conversion of fibrinogen to fibrin. So that is the formation of definitive clot. So this is in natural regarding the process involved in, and also you have clot retraction, clot lysis. So this is all about hemostasis, steps of hemostasis. Is there anything which is not clear for any of you? Any point need to be clarified?
Ma'am, 5-HT ഒന്നുകൂടി പറയോ? 5-HT.
5-HT serotonin. Serotonin വേറെ പേരാണ് 5-hydroxytryptamine.
Thank you, ma'am.
Okay, welcome. ഈ പറഞ്ഞ ഇത്രയും സ്റ്റെപ്സ് നിങ്ങൾക്ക് ക്ലിയർ ആണോ? ഞാൻ പറഞ്ഞതിനകത്ത് ഡൗട്ട്സ് ഉണ്ടോ? ഓക്കെ, അപ്പോ നിങ്ങൾ ഇതിനകത്ത് വേറൊരു കാര്യം കൂടെ മനസ്സിലാക്കണം. നമ്മൾ ഈ പറഞ്ഞപ്പോ എൻറ്റയർ കോയാഗുലേഷൻ പ്രോസസ് വെൻ വി ഹാവ് ബീൻ ഡിസ്കസിങ്, ഇതിനകത്ത് ഏറ്റവും ഇമ്പോർട്ടന്റ് ആയിട്ടുള്ള നിങ്ങൾക്ക് ഫീൽ ചെയ്ത കോയാഗുലേഷൻ ഫാക്ടേഴ്സ് ഏതൊക്കെയാ? Which is the most important? Yes, thrombin has got a, yes, 8 is very important. Yeah, 13. Calcium is important. Then
12. Factor 1 and 2.
1 and 2 are extremely important എന്ന് നിങ്ങൾക്ക് ഫീൽ ചെയ്തല്ലേ? ബാക്കിയുള്ള കോയാഗുലേഷൻ ഫാക്ടേഴ്സിന് കിട്ടാത്തൊരു ഇമ്പോർട്ടൻസ് ഫാക്ടർ 1-നും 2-നും കിട്ടുന്നതായിട്ട് നിങ്ങൾക്ക് തോന്നിയോ?
Yes, ma'am.
Yes, and also the thrombin ഒരു ഭയങ്കര പവർഫുൾ, means it causes activation of 5 to 5A, 8 to 8A, fibrinogen to fibrin, 13 to 13A. നിങ്ങൾ അത് ശ്രദ്ധിച്ചായിരുന്നോ? Factor 13 is, factor 2 is getting so much of importance, right?
Yes, ma'am.
So what kind of substance is that? Is it a procoagulant or an anticoagulant substance? Is it a
Procoagulant.
So far, as per our experience with this coagulation, so far what we have heard, we are under the impression that this thrombin is a very good fellow. He is a procoagulant substance. He will help us whenever there is an injury. He will cause clot formation, right? Yes, but there are some people, അല്ലെ, നമ്മുടെ കൂടെ ചില ഫ്രണ്ട്സ് ഒക്കെ ഇല്ലേ, ചില ഫ്രണ്ട്സിന് എങ്ങനെയാണ്, ആദ്യം നമ്മളോട് ഭയങ്കര കൂട്ടായിരിക്കും, പക്ഷെ കുറച്ചാൾ കഴിഞ്ഞ് വേറെ ഫ്രണ്ട്സിനെ കിട്ടുമ്പോ എന്ത് ചെയ്യും? സ്വഭാവം വേറെ ആളുടെ കൂടെ കൂടുമ്പോ അല്ലേ?
നമ്മളെ ചതിക്കും.
ചിലര് നമുക്കിട്ട് പണിഞ്ഞിട്ട് പോവല്ലേ ചിലര്. Okay, so you can compare your thrombin to those kind of your friends. It is like those kind of friends who will cheat you in between. Okay. So this thrombin basically it is a procoagulant substance. But at other times when you have sufficient amount of coagulation happened in your circulation, what the thrombin will do? Thrombin will act as an anticoagulant. Okay, so we will see what are the different mechanisms by which this clotting is prevented in a normal circulation. Normally നമ്മുടെ circulation at clotting ഉണ്ടാവാറുണ്ടോ? ഇഞ്ചുറി ഇല്ലാത്തപ്പോ? ഇഞ്ചുറി ഒന്നും ഇല്ലാത്തപ്പോ? No. Blood flows freely, അല്ലേ? ബ്ലഡ് ഇങ്ങനെ ഫ്രീ ആയിട്ടല്ലേ ഫ്ലോ ചെയ്യുന്നേ? ഇടയ്ക്കിടയ്ക്ക് ഈ കോയാഗുലേഷൻ മെക്കാനിസം ആക്ടിവേറ്റഡ് ആയിട്ടുണ്ടായിരുന്നെങ്കിൽ വാട്ട് വുഡ് ഹാവ് ഹാപ്പൻ? എന്ത് പറ്റും? ബ്ലോക്കേജ് ഓഫ് ബ്ലഡ് വെസൽസ് ഉണ്ടായ
Necrosis of tissues.
blood pressure.
Yes,
Die.
excellent. Necrosis of tissue. Tissue will be devoid of blood supply. The tissues will die. Is it a good thing?
No.
No. No.
Never.
So no, it should not happen normally. No. So in your body, you have a lot of natural anti-clotting mechanisms or anti-clotting mechanisms in vivo. Either ningal ezhudiyitto. This is a very, 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 very important exam question. This is a very, very important exam question. Natural anti-clotting mechanism. പല പ്രാവശ്യം പരീക്ഷയ്ക്ക് ചോദിച്ചിട്ടുള്ളതാണ്. എഴുതിയിട്ടാണേ, പിന്നെ ഞാൻ പറഞ്ഞില്ല എന്ന് പറയരുത്. Anti-clotting mechanisms in vivo. Okay, so there are a lot of complex mechanisms, interrelated mechanisms operate to maintain balance between coagulation and anticoagulation. ഞാൻ പറഞ്ഞു, you have a lot of procoagulant substances, you have a lot of anticoagulant substances, and in circulating blood, anticoagulants usually predominate, and there are certain factors which prevents blood clotting in a normal vascular system. What are they? One is the endothelial surface factors, second is the fibrinolytic system. Nammalipo fibrinolytic system already we have mentioned about that. Where have we discussed
Removal
about
of clot.
Yes, removal. Clot lysis varnappa, I have told you it is a fibrinolytic system which plays a role. Then you have the antithrombin action of fibrin, then antithrombin 3 and heparin. Then there is constant motion of blood also plays a role, and removal of activated clotting factors by liver. So these are the factors which normally prevents clotting in a normal vascular system. ഇത്രയും കാര്യങ്ങൾ പറഞ്ഞത് മനസ്സിലായോ? Okay.
Yes, ma'am.
So I think time is 11 now. It's about to get 11. So shall we stop here today? We have a few more slides to discuss that I will discuss in the coming classes. Shall we stop here today?
Yes, ma'am.
Yes, ma'am.
What class
Yes,
do you
ma'am.
have? What class do you have right now?
Ma'am, we have recorded anatomy, but it has
Anatomy.
been
Anatomy.
given.
Okay, so you do one thing. In the MCQs, actually, there is another important thing that you have to do. So, coagulation pathway, it's a very, very important topic for you. So, all of you, please draw the diagrammatic representation of the coagulation pathway. I'm sorry. Coagulation pathway. കേട്ടോ, എന്നിട്ട് നിങ്ങൾ അത് ഗൂഗിൾ ക്ലാസ് റൂമിൽ അപ്ലോഡ് ചെയ്യണം. ഞാൻ അത് അസൈൻമെന്റ് ആയിട്ട് ഇട്ടേക്കാം. That pathway is very, very important. You have to follow the diagram which is given in Genome. ഞാനിപ്പോ ഇന്ന് ക്ലാസ്സിൽ കാണിച്ച ആ പടം coagulation pathway. മനസ്സിലായോ ഏത് പടമാണെന്ന്? coagulation pathway.
Intrinsic and extrinsic അതാണോ?
Intrinsic and this pathway. Can you see the slide? This pathway.
Yes, ma'am.
Yes, ma'am.
ഓക്കെ, അപ്പൊ അതിൽ ഈ HMW എന്താണെന്നൊക്കെ സൈഡിലായിട്ട് എക്സ്പാൻഡ് ചെയ്തണം. TPL എന്താണ്, TFI എന്താണ്, അപ്പൊ അത് എക്സ്പ്ലെയിൻ അതിന്റെ എക്സ്പാൻഷനും എഴുതണം. ഇത് ഈ രണ്ട് പാത്വേ, ഈ കോമൺ ഫുൾ പാത്വേ, ഈ പാത്വേ SH അത് എഴുതി പഠിച്ച് നന്നായിട്ട് കാണാതെ ഒന്ന് എഴുതിയിട്ട് അത് എനിക്ക് അപ്ലോഡ് ചെയ്ത് തരണം. തരുമോ?
TFI ഫുൾ ഫോം എന്താണ്?
ടിഷ്യൂ ഫാക്ടർ ഇൻഹിബിറ്റർ.
Thank you.
Okay, tissue factor inhibitor or tissue factor pathway inhibitor. Is that clear?
എഴുതണോ?
ഡീറ്റെയിൽഡ് ആയിട്ട് എഴുതണ്ട, പക്ഷെ ഇതിന്റെ ഡീറ്റെയിൽസ് മുഴുവൻ നിങ്ങൾ നന്നായിട്ട് പഠിക്കണം കേട്ടോ. പരീക്ഷയ്ക്ക് ചോദിക്കുമ്പോൾ ദിസ് ഡയഗ്രാം alone is not sufficient. You have to elaborately write upon the intrinsic system, extrinsic system and the common pathway. Yes, this can come as a long essay. As I will give you all those questions at the end of the session.
Ma'am, could you once more tell what's TPL?
Pardon? Yeah, TPL. Is it TPL you are asking about?
Yes, ma'am.
Tissue thromboplastin. Tissue thromboplastin. Clear?
Thank you.
Okay. So shall we wind up our session today?
Yes, ma'am.
Yeah.
Yes,
Yes, ma'am.
Yeah, tell me. Yeah.
Yes,
Huh?
ma'am.
Pardon? So thank you all. Thank you all for your
Ma'am.
patient listening. Yeah. Tell me.
Can I ask you a question? Ma'am,
Yeah,
for
sure.
internal damage of the tissues or for internal bleeding also, will all these clot formation take place?
Yeah, sure.
Ma'am, then
You-
why do we tell ma'am like the internal bleeding or internal damage of tissues is more harmful than the external ones?
Yeah, because to your viscera, blood flow is very, very high, and also if there is any internal bleed, if the bleeding is external, it will be very visible to you and you will apply some tourniquet or some other measures to stop the bleeding. But if there is any bleeding internally, will you be aware of that bleeding?
No, ma'am.
Will you be able to see the bleeding? So will you be aware of the bleeding? The bleeding process will keep on continuing, and also this temperature within the system, core temperature is slightly high. That also inhibits clot formation. So clotting actually, it needs an ambient temperature, no?
Yes, ma'am.
Okay, so basically the thing is, like when you're saying like internal bleeding, what do you actually mean is that it's a bleeding that occurs within the viscera, like your organ, like within the stomach or within the any kind of tissue within. So in those organs, like if stomach or liver or something like that is involved, the blood flow to those tissues are very high, to the kidney and all, the blood flow is very high compared to your skin.
Oh, okay, ma'am. Yes, ma'am.
Okay.
Yes, ma'am.
Okay.
Thank you.
Anything else? Anything else? Anything else? Shall we stop here today?
Yes, ma'am.
Yes, ma'am.
Okay. So thank you all for your patient listening. With 2 more classes, we'll be finishing platelets and hemostasis. So see you later. Thank you all. Thank you. Bye. So my classes will be I think the next classes will be on Friday or Saturday, it seems. So anyway, you just be prepared for the sessions. We'll see you in the next class.
Okay, ma'am.
Thank you.
Thank you.
Thank you, ma'am.
Bye-bye.
Thank you, ma'am.
Excuse me, ma'am.
Yeah. Tell me.
Ma'am, will the next class be based on the same PPT of class 3 platelets?
Yeah, the remaining things of this slides, along with I will upload the slides for next session.
Ma'am, for the next session, do we have to only see this slide?
Pardon?
Ma'am, for the next class, do we have to see the part 3 slides only?
No. Additional slides will also be there. In the part 3 slides, we have only a few slides left behind. Along with that, the other slides also we'll have to go through. I'll upload those slides.
Okay, ma'am. Thank you, ma'am.
Thank you. Bye.